Okay, just a brief introduction to what we're dealing with today. I don't think I need to tell anyone nowadays about the importance of disease surveillance. This has risen high up the agenda, but of course, many people perhaps don't think that it's relevant to plants as well as to animals. So these are three of the diseases that we're going to be dealing with in the survey. Yellow rust on the left, brown rust in the middle, and powdery mildew on the right-hand side. The programme today will include comments on other diseases, but the main focus of the survey obviously is on these. Next slide. Now, there will be some talks dealing with disease management, and there are a number of options available to growers, which I'm sure you'll know well. There are some agronomic issues, for instance, sowing date. Use of resistant varieties, of course, is a high priority, and we will be talking about the recommended list in the programme. Fungicides, of course, are still widely used. I put a question mark against biocontrol because at the moment on cereal crops, the biological control options seem to me to be relatively limited, but that hopefully may change in the future. And importantly, there are, of course, some constraints. We've got good tools, but there are constraints. And the main one is that pathogens are a moving target. They evolve over time for both virulence towards different varieties and, of course, towards the fungicides we use, resistance. And one of our talks will be dealing with that topic in particular. Next slide, please. Just to give you some sort of indication that the plant breeders have been busy trying to improve the situation in terms of selection of varieties. This is a slide I show each year, but I update it. So if you look underneath there, you've got the 2021, 21-22 and 23-24 recommended list ratings for yellow rust. And I think it's quite encouraging if you look at the progress towards the right-hand side of the screen, that the number of resistant varieties obviously mainly in adult plants rather than seedlings, I think, has been increasing over time. So the choices are improving. Of course, this doesn't mean to say that these varieties will necessarily have those high ratings indefinitely because of what we talked about previously with the emergence of new virulent pathotypes. But nonetheless, it's quite an encouraging picture. Next slide, please. Okay, just to finish my comments, an overview of the programme. The main part of the early morning will be the reports from the 2022 season. Obviously, the focus is on the survey there. Then we will be having our break. After the break, there's a range of different talks, one on integrated pest management from Lisa Jorgensen, then the presentation mainly dealing with the evolution of fungicide resistance from Nicola. Paul Nicholson will be talking about Fusarium head blight, and Aoife Driscoll will really be talking about how some of the more fundamental information that we have on pathogen populations can be put into practice. And then we're going to conclude appropriately with comments on the recommended lists and the actual survey itself. And then we're going to have a short opportunity for final questions, and then we're going to close the meeting at 1:30. Okay, so that's me done, and I think we can move straight on to our first presentation, which is from the UKCPVS team, starting with Charlotte. Charlotte is actually the programme leader in pathology at NIAB and has interest in disease-resistant characterisation, and she's the lead scientist for the UK Cereal Pathogen Virulence Survey. Amelia Hubbard, our other presenter, is another plant pathologist at NIAB, and she's responsible for a lot of the actual practical work on the survey, all the kind of disease screens and testing. So I'll hand over at this point to Charlotte.
Thank you, John. Oh, sorry, I've skipped ahead. Okay, so welcome to the webinar this morning. So this is the programme for our presentations on the UK cereal pathogen virulence. survey. So I'm just going to go through some introduction slides, and then Amelia will take over and she'll talk through all the data from the 2022 season from all the different pathogens listed on the screen. I'll then take over and talk a bit about sampling in this current season in 2023, cover some of the main take-home messages, and then we'll have some time hopefully for some questions at the end. So just for those people that are not aware about the survey, this survey, so the UK Cereal Pathogen Virulence Survey, was established in 1967, so it's been going for 55 years. And this was initiated following an outbreak of yellow rust on the previously resistant variety Rothwell perdix. And the aim of the survey is to identify changes in pathogen populations and detect new races that may have adverse effect on cereal production in the UK. We rely on farmers, agronomists, trial staff, breeders and researchers to send in infected leaf samples, and we're grateful for every sample that's sent in to the survey. The survey is funded by AHDB and also AFA, the Animal and Plant Health Agency. So the current pathogens that were surveyed in 2022 are wheat yellow rust, shown on the left-hand side, wheat brown rust in the centre, and then the wheat and barley powdery mildews on the right-hand side. And I just wanted to go through the process of how we deal with the samples. So this is particularly focused on rusts. So we receive samples into the survey. They're first registered on our database, and the leaf sample is placed into incubation, and this is so that the pustules start to sporulate. We then transfer these spores onto a susceptible variety, and after about 14 days we can start to see developing pustules producing spores. So we start to collect these off these susceptible plants, and this can take several weeks. Sometimes we may need to repeat the process over several successive rounds so that we can multiply enough spores to work with. And then from these initial pots, we can take a segment to store at -80 so they can be genotyped or and stored for later use for genotyping. And at the end of the season, we select 25 isolates of wheat yellow rust and 25 isolates of wheat brown rust to go onto the seedling differential screens. And I will mention a little bit more about that on the next slide. And we aim to complete these tests by the end of December. From these 25 isolates, we select 5 isolates of yellow rust and 5 isolates of brown rust, which we take on to our adult plant tests in the field and also our variety seedling tests in the growth room. And these tests assess the risk of those population changes on our UK recommended list of varieties and candidates. And finally, once the all isolates go into long-term storage, and so we're not able to test all of the isolates that are sent into us, but we do store everything that is able to be multiplied. And we have a collection dating back to the 1970s, and there have been times where we've gone back into this collection to have a look for and use older isolates. So this is just to highlight that if your isolate isn't selected for further research, it is still stored and may still be useful later on. So if we look at our seedling differential tests, we use these to monitor pathogen populations. So we're able to determine which virulence genes the isolates carry. So this is because single major resistance genes, which can be used in varietal resistance, can be overcome by new races in a short period of time. So our work helps us keep track of of these diseases, and often gives us the first sign that the pathogens are evolving and adapting to resistance genes in our UK varieties. So this table just shows how our tests, or gives a bit more information about our tests. So we have the differential collection contains these differential cultivars, and these have known virulence genes. So for instance, if we just look at Chinese 166, it has the resistance gene YR1. So if an isolate of yellow rust is able to infect Chinese 166, then we say that, then we know that this isolate carries the virulence gene for YR1. So it's able to overcome this resistance. And we use this information across all the different differentials to build up what we call a virulence profile, which we term a pathotype. So this just gives us the collection of virulence genes that the isolate carries, and also any additional test cultivars that we have in our test. And this is this is to show infection at the seedling stage. So what you'll see is this pathotype, an example pathotype on the screen. So we have the first part of it is the collection of the virulence genes, so the numbers, and then the additional cultivars are shown and in the right-hand portion of that code. And this is just so that we can understand the differences between the different isolates that we obtain from the survey. So our current naming system uses a combination of colours and numbers. So this reflects the complexity of the yellow rust population. So we use the colour code to denote the genetic group. So for example, here we've got the red group, which is very diverse, and the number is allocated to the pathotype, so that combination of those virulences that the isolate carries. So for example, here we have red 28, which the pathotype is shown on the screen. And for the red genetic group, there are over 50 red pathotypes. So this genetic group is very diverse. We take 5 individual yellow rust isolates and 5 individual brown rust isolates onto adult plant trials. And this is just an example of how we set out these experiments. So they're hand sown in these tussock trials. So for each of the individual isolate, there'll be an individual tussock setup. And in the picture, you can see how it's laid out. So we have 2 reps of these hand-sown tussocks with varieties in a randomized design. We erect the windbreak around the trials to help protect the plots and aid rust development. Sorry, I should have clicked on these buttons. And then I just wanted to show the pipeline of how we deal with these samples. So I've tried to explain it on the previous slides, but I thought perhaps having this pipeline may help. So we receive the samples. For instance, last year we received them across the summer of 2022. We spend the summer reviving and bulking these isolates. And as mentioned, we select 25 isolates, which we take onto these differential seedling tests. And as mentioned, this determines the pathotype of the isolate on characterized and uncharacterized resistances, which we term differentials. And this happens across the autumn of 2022, for example. And then these 5 isolates are selected, which go on to the RL variety and candidate seedling tests, which are done sort of winter, spring of 2023, for example. And these results from the seedling tests are now incorporated into the RL table, so under the young plant rating. I'll mention a little bit more about this on a later slide. And then we also test these 5 isolates on the adult plant trials, as mentioned. And this data that we get from the adult plant trials is used to validate the watch list. So why are adult plant trials important? So there's different stages of resistance. So we have seedling, which is also called all stage resistance, and it means that the host is usually resistant at all growth stages. But we do have adult plant resistance, which kicks in at a later growth stage. And so therefore, just because something is susceptible as a seedling, it doesn't mean it will be susceptible as an adult. And as we all know, high disease levels can affect yield. So using susceptible varieties can affect on-farm inputs, as it's more difficult to control the disease. So variety choice can limit or increase the spread of disease. So UKCPVS helps to provide more info to breeders to help them develop new varieties. And as pathogens are constantly evolving, so it's important to monitor how individual races and isolates affect varieties. So AHDB run inoculated trials as part of the RL using a mix of isolates, but what's different about UKCPVS is we look at how these individual 5 isolates of yellow rust and brown rust affect varieties. And so I mentioned about the young plant resistance in being used in the RL. So in the new booklet, you can find it on page 10. So you'll see, as highlighted in the green, this line here is now included, and the data's listed as either an R or an S, so for resistant or susceptible. Catherine will explain a bit more about this in her presentation later this morning, but please do use that information in the booklet. I'm now going to hand over to Amelia, who will go through the data for the 2022 season, starting with wheat yellow rust.
So good morning, everyone. My name is Amelia Hubbard, and I'm here to show you the wheat yellow rust. I'll just start my slideshow. So I'll be going through all the diseases today, starting with wheat yellow rust. So before I start, I'd just like to point out the letters in the top right-hand corner, so you'll always know what disease I'm currently talking about. So we have WYR for wheat yellow rust, and then when I go on to the wheat brown rust, it will change to WBR. So firstly, we're going to start with the wheat yellow rust adult plant trials. So these were the ones that were in the ground in 2022, and they were inoculated with isolates from the 2021 survey. So if we have a look at the isolates that we used 2112. That one was from KWS Chilham, a spring wheat, and we selected that one because it was from a common pathotype in 2021 called Red 36. We selected 2114 from LG Skyscraper, and that one was selected again because it had another common pathotype that year called Red 41. We selected 21045 from Skyfall. That one was interesting because it had virulence for YR8, and we don't see that one very often. And in its differential test, it infected both our YR8 differentials for so Avocet YR8 and Compare. So we took a look at that one. We also selected this one for 21102. That was from KWS Jackal. So that one also had virulence for YR8, but it combined it with virulence for Crusoe, and that was a new pathotype that year. And we selected this one, 21135 from RGT Wolverine. It had a pathotype for Red 11, and we picked that one because I was also using it in some other tests, and it was looking interesting at the time. So it's just trying to choose isolates that are going to give us the broadest picture of the isolates we received. So these are the results of the adult plant trials. I'm not going to go through every variety because obviously there are a lot, but I'll take you through some of the results, and I've expressed it as a mean percentage plot infection over 4 assessments, and these assessments started at growth stage 50. Each colour on your charts represent a different isolate. So for blue, we have the isolate 2112, and then in green, we have isolate 2114, and so on. So if we take Robigus first, that's one of our control varieties, and it was the most susceptible variety in the yellow rust trials. And you can see it was susceptible to all 5 isolates at adult plant stage at around the 26 to 53% mark, with slightly higher levels with 2112 and 2114 in the blue and the green. In the middle, we have Cadorus, which had a rating of 4 last year, and it was the most susceptible RL variety in trials. And you can see we did have some differences between the isolates here, and it was more susceptible again to 2112 and 2114. And the last one is Skyfall, which had a rating of 3, and that was again susceptible to all 5 isolates at around the 20 to 30% mark. So if we have a look at some of the other varieties we included, so we have RGT Wolverine first. I should point out that I've changed the scale on the y-axis here, so we were going up to 53% on the first slide, but now we're only going up to 20%. So Wolverine was infected in all 5 trials at around the 9 to 20% mark. Gleam with a rating of 5, that saw quite low levels, slightly higher with the first 2 isolates, and we would have normally expected to see probably a bit more on Gleam, but I did find that in these inoculated trials last year that the infection was held back by this very dry April we had, and then the warmer temperatures, which seem to impede development of the rust. So I think it's more environmental conditions, because I did inoculate, these are heavily inoculated plots, and they would have been inoculated weekly for about 8 weeks. So it's not for want of trying, but we're trying to aim for high disease pressure in these trials. If we take Swallow in the middle there, so I've just picked out Swallow as to represent the varieties rated 6, and you can see, yes, we saw an infection with all 5 isolates, but at really quite low levels of between 1 and 3%. RGT Stokes with a rating of 7 last year, again, very small trace amounts with some of the isolates. And then we have Champion Lars with a rating of 8, and again, just a smidge with 2 of the isolates there. So you can see we had this general pattern where as the rating increases, the amount of infection we were seeing decreased, and that's not unsurprising, and it's what we expected to see. So we did actually have quite a few varieties that were resistant to all 5 isolates, and I've picked out the ones that had 0.1% or less infection over those 4 assessments. And there's 22 varieties here from the RL. I haven't picked out the controls, I've just picked out the RL varieties for you. And the ones with an asterisk are the ones that were completely resistant, and we didn't see any infection at all on those. But they're all classed as resistant due to the low levels. And if you're interested in a variety that I haven't talked about here, then please do pop along to the AHDB website, because our annual report is on there, and it has the full results on there for you for last year's adult plant trials. So as well as our initial seedling differential test, we also perform RL variety seedling tests on our just using those 5 isolates that we selected for adult plant trials. And I've compared just some of the results here for the seedling stage alongside the adult plant reaction. So where you see an orange colour on the left-hand side of the table, that denotes an intermediate reaction. So we class it as a borderline reaction of between 2.4 and 2.6. And anything scoring 2.7 and more is classed as susceptible, and it's a 0 to 4 scale. For the adult plant, I've merely highlighted everything in red that scored over 10%, just so we could see it a bit more clearly of those higher figures. So if we just have a look at some of these results, the ones in green here all rated 8 or 9 last year, and you can see they were all resistant at seedling stage and resistant at adult plant stage. So these have good all stage resistance. If we have a look at the ones in the blue box, so we've got Cadorus Jackal here, Elation, Illicit and Graham with ratings of 7 and 8. You can see that these were susceptible to 2 or more of the isolates at seedling stage, but again were resistant at adult plant stage. So these ones were showing adult plant resistance which kicked in at later growth stages. And then at the bottom we have RGT Wolverine, Skyfall and Zayat, all rated at 3 or 4 last year. These ones were susceptible at seedling stage and susceptible at adult plant stage, although you could argue that RGT Wolverine was sort of like intermediate with 2145, but generally those levels were higher. And of course we've got Zayat at the bottom there as our most susceptible RL variety in trials. So as well as our field trials, we wanted to see if we could kind of like produce those results for you sooner. Can we do it in sort of like controlled environmental conditions to produce these results instead of waiting for the field trials, which are quite lengthy? You drill them in the autumn, you have to inoculate them in the spring, and then wait for the summer to start scoring them. Can we do it in our glass houses? So we had a go, and these were grown under what we're calling extended day length conditions to really speed them up and rattle them through those growth stages. We picked 2 isolates from the 2021 survey, and we picked 2114 and 21045. And we chose 15 varieties with a range of resistance rating, including 2 control varieties. And because these weren't plots, they were individual plants, I assessed them on leaf layers. So I'd done 3 leaf layers, and I assessed them twice. But we did have some challenges with this work. Rust doesn't really like extended day lengths, so we chose to move the plants to optimum conditions for rust development after we inoculated them. So we have the little picture of Caderus barrel there, just to highlight this is what I was aiming for. I inoculated them at growth stage 39 because I wanted to get that adult plant reaction on the flag leaves. So I just thought I'd show you a picture of the one of the results. So these are the results with the varieties that we chose. So again, just to point out, it's a percentage leaf area infected, and we have the scores here for leaf 1, 2 and 3, where the flag leaf is leaf 1. And it was an average of 5 leaves over 2 assessments. So I hope you can see that. I'll just pick out some of these varieties. So Crusoe rated 9 last year. You can see that it had 12% average infection on L1 with the isolate 2114, but it was only at 2 or 3% with 21045. Siskin, Theodore and LG Astronomer. So these ones were actually resistant to both isolates. Siskin was particularly resistant. I didn't see any yellow rust at all on Siskin, but Theodore and Astronomer did see some very low levels of 1 or 2% on some of the leaf layers. Caderus stays with a rating of 8. You can see that one was susceptible to 2114, but really quite resistant to 2145. Caderus Skyscraper with a rating of 7. You can see slightly kind of similar to Extays, where it's more susceptible to one isolate compared to the second isolate. Caderus Barrel and Caderus Firefly, so I rated these, or lumped these in the same box because they both got a rating of 6. And you can see out of the 2, it's Barrel that was more susceptible than Caderus Firefly. Gleam and SY Inciter with ratings of 5. You can see we were seeing up to 30% on leaf 1 for the isolate 2114, but quite variable levels with the isolate 2145. Caderus Zayat and RGT Wolverine. Again, I've lumped these all together as susceptible varieties, but we did see quite varying reactions. We had up to 44% on leaf 1 with 2114. And then you can see, if you look at the Skyfall, we have up to 52% on L1 with the second isolate. And our 2 control varieties. So you can see I've got Rhobagus in grey, and that's because unfortunately in one of the experiments Rhobagus failed. I don't think it really liked the speed breeding conditions, and I did have a couple of problems with it, so I couldn't get any scores off that those pots. And VUCA, you can see that one's in grey for leaf 1, and that's because VUCA didn't actually reach growth stage 39 by the time I did my first inoculation. And although I did inoculate them twice, the flag leaf had only just come out, and by the time I'd finished scoring, it still hadn't developed. But you can see from the other, if you have a look at VUCA with 2145 on leaf 2 and leaf 3, I can see that the inoculations did work because we were seeing up to 58 and 72%. So just a summary slide for the off-season adult plant trials. So some of our control plants failed, and that's due to either a later growth stage at the time of inoculation or stress under those extended day length conditions. Cadosiskin was the only variety to remain completely resistant to both isolates, so not a whisper on Cadosiskin, but Theodore and LG Astronomer were also resistant to both isolates with very low levels of infection recorded. And Cadivus extase, that one was susceptible to the isolate 2114. So we have some pros and cons of this type of experiment. So my pro is that yes, we can deliver that adult plant data ahead of the season. So we could be doing this in the winter and get those results to you much sooner than a field trial. But the cons are that we have a forced growing condition, so that may affect the growth of the plants, and the results were not comparable in our trials to the actual results we were seeing on the 2022 adult plant field trials. And it's expensive. We only did 15 varieties and 2 isolates, but I put each isolate trial in a different growth room. So that's 2 growth rooms, and if I'd done the full set of RL and RL candidates against 5 isolates, like I do in the field, then it would take up a huge amount of space in either a glass house or a growth room. So now we can move on to the 2022 samples that we received for wheat yellow rust. So we received 187 samples. They came from 27 counties and across 52 varieties. The most sampled variety was Calibus Sciate, and that was closely followed by Skyfall. So we had 16 samples in from Sciate and 15 samples in from Skyfall. We had 4 samples in from Siskin. We had 2 from Costello and 2 from Mayflower, and 1 from Theodore, and they were all rated 9 last year. And we selected 25 isolates for our yellow rust seedling differential tests. So I want to just have a look at the sample intake. So if you have a look at the green and the blue lines, you can see that our busiest month is usually June. That's when we receive the most samples. So this just is the total number of samples we received each month. And if you have a look at the orange line, that's for the sample intake for 2022. And you can see by the end of April, I hadn't even received 20 samples, which is quite unusual for us. We usually have quite getting up towards at least double that in April. So we did put a call out on social media, and that gave us a boost to our sample numbers. But you can see we also had this shift where May last year was my busiest month for samples, and we received over 120 samples in May. I didn't get as many samples in June that I would normally get, and we didn't get any in July, but we don't normally see get many samples in July. But it did seem that the whole shift of the epidemic did, it shifted back a month for us. And I think that's due to the warm weather and the fact that the crops were ahead of schedule last year. So they certainly were for my trials. I did go back and check, and I noted that even in my inoculated trials at growth stage 59, they were about 12 days ahead of what they would have been the previous year. So I'd like to go through the yellow rust virulence frequencies. So these are the seedling test results, and this chart is the percentage of isolates carrying virulence for the main resistance genes. So very quickly, YR 1, 2, 3, 4, 6, 7, 9, 17, 25 and 32 all remain very, very high in the population, as it's been seen in previous years. So we've got the data for the last 5 years here, but it was virulence for YR8 that we were particularly interested in, and I didn't detect any isolates from 2022 that had virulence for YR8, so that disappeared. And I should point out that no virulence was detected for YR5, 10, 15 or 24 either. If we have a look at the virulence frequency for the additional cultivars, just some of them that we also include in the test. So we have virulence for cadenza. That one has been increasing slightly over the last few years, but in 2022 it returned to its 2018 level, so I don't think that's anything to worry about. Virulence for Apache dropped slightly from what we were seeing in 2021, but again it's very similar to previous years. Virulence for Evolution in the middle, that one did peak in 2020 where it was seen in over 70% of isolates, but in 2021 and 2022 it's remained at around the 40% mark. And then virulence for Chronic, so that one wasn't seen in 2018, so it's missing that bright blue bar. We did see it in around 40% of isolates in 2021, and then it's dropped back down to around 20% for 2022. Virulence for Crusoe at the end there, that one again wasn't seen in 2018, so no bright blue bar, but it was seen in around 8% of isolates in 2022. So these are the pathotypes that we found in 2022. So that's another name for the virulence profile of these isolates. So the most common pathotype that we identified was the one for red 26, and that was in 28% of the isolates. So we've seen red 26, it was the first identified in 2016, and it's been seen most years since, so we weren't surprised to see that one. Red 24, that was in 20% of the isolates, and that one again was first seen in 2016, but it was the most common one in 2017, and it did cause quite a lot of damage to our RL varieties back in 2017. So that one, it's also been seen at very low levels since then, but it was the second most common pathotype in 2022. So we did have some new pathotypes. We had red 84, which was only in 4% of isolates. So that one was new because it had a virulence for YR4, so it didn't infect our YR4 differentials. It was avirulent on Rendezvous and avirulent on Warrior. And you can see the 32 is in brackets. That's because we have two YR32 differentials in our tests, and it only infected Avocet 32. It didn't infect Carstens 5, and usually virulence for those two goes hand in hand. And I did actually retest that isolate just to check that result, and it didn't infect Carstens 5 in its second test either. Another new pathotype was for red 85, so that one was new because it had this combination of virulence for Kelleria sterling, which is denoted by ST, Chronic, Apache and Evolution. And we had pink 18 as a new pathotype. So that one was interesting because it had virulence for Crusoe. So the genotyping of wheat yellow rust. So our aim is to conduct routine genotyping of wheat yellow rust isolates using results from Diane Saunders from JRC's Field Pathogenomics project and the Mobile and Real-Time Plant Disease Diagnostics, or MARPL, pipeline to categorise isolates into the different genetic groups. So NIAB is optimising the genotyping here, and we've now genotyped 24 isolates from 2019, 24 isolates from 2020, 48 isolates from 2021, and we've done 24 isolates from 2022. So I just wanted to show you the genotyping process. So we actually extract DNA from the leaf samples, and then we use PCR to amplify 242 genes. These are then pooled, and the libraries are sequenced on the GridION, and the GridION uses long read sequencing technology. And then we can use, we can produce this phylogenetic genetic diagram, and there's a simplified diagram of that on the bottom left-hand side of your slide. And I'd like to point out that it's Helen Bates and Anne Webb here at NIAB that do the genotyping. I don't do it myself here, too busy doing all the practical work, and so thanks to my colleagues for their help with this. So the MARPL genotyping, it actually separates the isolates into the genetic groups. So this is the phylogenetic tree. I won't go into too much detail of this because it's a huge diagram and it's difficult to display on these screens. But basically, the middle of that diagram is the family tree, and then the outer ring is the results from the seedling test, so that's the virulence profile of the isolates. But the main take-home message from this slide is that the red group has dominated for the last 4 years. So in 2022, out of those 24 isolates that we genotyped, 23 isolates belonged to the red group, and just 1 isolate was in the pink group. And that we've actually noticed that's kind of the same situation that they've even been seeing over in Europe with their genotyping. The majority of their samples belong to the red group with a small proportion to the pink group. And they do have some other groups over in Europe, but we don't see them here. And the red group, it's huge. It's a huge group. It contains many different isolates and a broad range of virulence profiles. So over the years, it has helped us to ID these unusual isolates. So in 2019, we had 1 isolate in the purple group, which was 19185. The purple group, if you've been following the UKCPVS for a long time, you may remember that we used to call this one the chronic race. And then at the bottom of that little diagram, we had the 5 isolates in the pink group. So we had 2 from 2019, 2 in 2020, and just the 1 in 2022. The pink group, again, you may remember that is called the warrior group. And if on that diagram, you can see that our original warrior race, which was isolate 1108 for warrior, that one did fall into this pink group here. So it's like they've all lumped into that one area on the phylogenetic tree. So just a quick look ahead at the 2023 isolates that we've chosen to for this year's adult plant trials. So we've chosen this one here, 22038 from Costello. That one's been chosen because it had a pathotype called Red 76. Red 76 was found in 2021 in just 1 isolate. We didn't select it for adult plant trials last year, but that pathotype cropped up again in 2022. So that isolate has been selected for adult plant trials this year. We selected 2255 from Gleam. That had quite a simple pathotype. With the yellow rust, we quite often see these broad pathotypes, and with many infecting many of our additional cultivars. And this particular one from Gleam only infected Spalding's Prolific, Robigus and Solstice for the additional cultivars. So we picked that one there. We selected 22143 from Mayflower. So that was one of our new pathotypes with the 32 in the brackets and the A virulence for YR for Rendezvous and Warrior. We selected 22162 from Cadwis Siskin. So that's our pink isolate, the only one that we identified in 2022. And that particular isolate had virulence for Crusoe. And we selected 22180 from Theodore, and that one was selected because it was the most common pathotype in 2022. So a summary for the wheat yellow rust. The varieties generally performed as expected in adult plant field trials. Many of the 2022 RL varieties were resistant to all isolates at adult plant stage. Infection levels may have been reduced due to the weather. We received 187 samples in 2022. For us, the epidemic peaked in May. No virulence was detected for YR5, 8, 10, 15 or 24. The most common pathotype was this one here, belonging to red 26. We had 3 new pathotypes identified, and the genotyping showed that 23 of the isolates genotyped were red group isolates, and there was 1 pink group isolate. So I'm going to move on to the brown rust just because I am conscious of the time, but we will be taking questions at the end. So this work was previously done by Sarah Wilderspin last year, but she's since left NIAB, so I'm afraid it's just me again for the wheat brown rust. So we're going to start the brown rust with those 22 adult plant trials and the isolates that we chose for those inoculations. So we chose 21002 from Caris Kerin. So for the pathotype, you can see that we have some different numbers there, and that's because with the yellow rust, we were talking about YR genes, and with the brown rust, we're actually looking at LR genes. So brown rust, other name is wheat rust, so it's LR genes. So, and some of these LR genes combine letters and numbers. So going back to this 21002, we selected this isolate for our inoculations because it was from Kerin, which was rated a 7 last year, but this pathotype had virulence for LR2C, and we don't see LR2C that often. We selected this one from 21003 from Skyscraper, and that one was the most common pathotype in 2021. We selected this one here from number 21004 from Kedros Bassett, and that one was selected because it was the second most common pathotype in 2021. We selected this one here, 21006 from Kedros Cranium. So that one was a new pathotype. It combined virulence for LR20 and 28. Again, 20 is in brackets because it didn't infect all of the differentials that we have for LR20, but pretty sure it does carry LR20. It was the only sample we had in from Lincolnshire as well, so we selected that one. And we selected 21008 from Relay, and that was a new pathotype in 2021. So the wheat brown rust adult plant trials, again, this is a percentage plot infection over 4 assessments, and these assessments actually started at growth stage 55. So the first variety I have is Crusoe, with a rating of 3 last year. So that one was actually the most susceptible variety in trials, and the most susceptible RL variety in trials. So it even beat some of our control varieties like Buster. It was infected in all 5 trials at around the 25 to 35% mark. I picked out Cados Guiam with a rating of 3 last year because again that was susceptible in all to all 5 isolates, slightly more susceptible to the isolates 21006 and 21008 in the blue and the red colours. Champion with a rating of 5, that one was susceptible to all 5 isolates and slightly more this time to the isolate 21003 in green and again 21008 in red. And again I picked out RAGT Rashid to represent the varieties rated 6, and you can see that one was susceptible to all 5 isolates, but at around the sort of like the 10 to 15% mark. And again, we see, like we did with the yellow rust, a pattern where as the resistance rating increases, the levels of disease we were seeing decreased. The next set of varieties I'd like to show you. Please don't be alarmed by RGT Saki looking rather worrying there with those high bars. That's because, again, I've had to change the Y axes for it. So RGT Saki with a rating of 7 last year was infected in all 5 isolate trials at around the 7 to 11% mark. LG Astronomer with a rating of 8. So that one was actually, although these levels are sort of like quite low infection, so it had around between 3 and 7%, it was still showing higher levels of infections than other varieties with the same rating. And I've shown them some of the other varieties here. So Next door to it is RGT Wolverine with a rating of 8, and you can see RGT Wolverine had very low levels of infection of only up to 2% with that 21008 isolate. Theodore, really not much, just a smidge with some of the isolates, and around the sort of like 0.5% with just the middle isolate 21004. And Skyfall was our most susceptible variety in trials with just trace amounts to those isolates. So going on to the RL variety seedling tests for brown rust and comparing them with their adult plant reaction. Again, for the seedling test, the orange colour, that's the borderline reaction. We're really looking at the reactions for 2.7 and over, as it's just 0 to 4 scale. And the adult plant, again, I've just picked out the varieties that scored over 10% in percentage plot infection, just to highlight them so that to make it more easier to read. So if we have a look at the first two, we have Skyfall and Theodore, our most resistant varieties in the adult plant trials. You can see Skyfall was actually susceptible to just one of the isolates at seedling stage, but resistant at adult plant stage. Theodore was completely resistant to all 5 isolates at seedling stage, and again, resistant at adult plant stage, so that one had good all stage resistance. RGT Wolverine, that one was susceptible to all 5 isolates at seedling stage, but resistant at adult plant stage, so that resistance must have kicked in at later growth stages. I've put a blue box around these just to highlight some of them. So these had a range of resistance ratings last year, and they infected 2 or more of the isolates at seedling stage, and you can see that the adult plant reaction did vary, with the ones towards the bottom of that blue box having the higher levels of infection. And I just wanted to point out Cados cranium here with a rating of 4 last year. You can see it was actually more resistant in our field trials compared to quite a few of the other varieties with higher resistance ratings. And finally, we have Crusoe at the bottom there, our most susceptible variety in trials, susceptible to all 5 isolates at seedling stage and susceptible as an adult plant. And again, if you're interested in those full results, they can be found on the AHDB website on the UKCPVS page. So our 2022 brown rust samples, so we received 28 samples from 8 counties and across 24 varieties. Unusual brown rust sightings, well, we did have a report of Theodore in Devon being infected at around the 30% mark, but we didn't unfortunately receive a sample from that. I did receive a sample from Theodore in Dorset, which was coded 22024, so basically it was 24 samples that came in last year, and I will be talking more about that one in the coming slides. And we selected 25 isolates for the testing on the brown rust differential set. So again, the brown rust sample intake, I wanted to show you this slide because although we don't really get that many brown rust samples in each year, June and July usually tend to be our busiest months. And usually July just beats June in terms of number of samples. But if we have a look at 2022, which is now in the dark brown colour, you can see that it had a shift again, very much like the yellow rust. July was no longer our busiest month. In fact, I didn't even receive any brown rust samples in July. June was my busiest month for brown rust samples. And we even received some samples in May and April, which we don't usually see samples in those 2 months for brown rust. So there's it mirrored what happened in the yellow rust epidemics. So these are the brown rust seedling test results from our differentials, and again expressed in terms of percentage of isolates carrying virulence. So a lot of them return to levels seen previously. So virulence for LR3A, LR3BG, LR3KA and LR23 all return to levels seen previously. So we're now looking at the green and the brown bars. so levels that were seen back in 2019. I have picked out some of them here, which I thought were more interesting. So we have virulence for LR2C. That one has continued its decline. It peaked in 2018, but it's been seen at quite low levels since then. We have virulence for LR24. So that's our new thing with brown rust for 2022 isolates. We saw virulence for LR24 in 12% of our isolates. And virulence for LR28 has continued to increase. I didn't want to chart all the resistance genes, so I've just got a couple of bullet points for the other ones. So virulence for LR1, 10, 13, 14A, 15, 16, 17, 17B, and LR37 was found in 100% of isolates, and that generally follows the pattern of previous years, at least the last couple of years. No virulence was detected for LR2A or LR2B in 2022, and LR2A remains undetected in the UK population since we've been including that diff since 2016, and LR2B hasn't been seen since 2017. So I wanted to go back to LR24 and look at that one in a little bit more detail. So virulence for LR24 was last seen in 2017. And then 3 out of our 25 isolates, that's 12%, infected LR24 in 2022. The additional cultivars, Stig and Warrior, were also susceptible to all 3 isolates, and they are thought to carry the LR24 resistance gene. And this might explain the high level of brown rust seen on Theodore in Devon, because one of the LR24 isolates, that 22024, was actually from Theodore in Dorset. And I checked back on the notes, and the sampler did note that infection, although he was scoring the infection levels at 3% overall, he also noted 10% in foci. So, you know, 10% on Theodore, that's a lot for Theodore. So I think this may be similar to that one that somebody had also seen in Devon. So that brown rust isolate 22024 has been selected for inclusion in the RL-VL isolate mix for the RL-VL inoculated trials. If you haven't heard that the NL national list is now being called the variety list, that's why it's VL. So yeah, we're including that one as it's an interesting isolate, and it will. So the inoculated trials that we perform for the RL and the VL will have that isolate in there in the mix. And I just thought I'd on my map show you where the other 24 LR24 isolates came from. So KWS Extase and KWS Ultimatum from 2 sites in Cambridgeshire. So it was spread across the country and across different varieties. It wasn't just a localized event. So now we can look at the brown rust pathotypes found in 2022. So we had 2 common pathotypes at 20% frequency and 16% frequency, and they've both been seen before. And we had some new pathotypes for the brown rust. We had 12% of isolates carrying that virulence for LR24, and all 3 of those isolates showed the same pathotype. We had this new pathotype here with virulence for LR20, 23, 26, and 28. We had this one here, the new pathotype for LR2C. So, yeah, we've seen LR2C before, but it's that particular pathotype that was new for 2022. And this one was new in 2022, where it had a virulence for LR3BG. So just to look at the isolates that we've chosen for this year's adult plant trials, we've chosen 22006 from Siskin, and that's the pathotype that had LR2C. We chose this one from 22014 from Crusoe, and that one was chosen because it was a new pathotype in 2022. We chose this one from 2218 from Oxford, and that one was chosen because it was the second most common pathotype. And we've chosen this one, 22024 from Theodore, because it's one of those LR24 isolates. And we've chosen this one here, 22026 from Mayflower, and that was one of the most common pathotypes. So we've picked a range of isolates showing the different types we were seeing last year. So a summary for the wheat brown rust. Varieties performed as expected in adult plant trials based on their RL rating. 28 samples were received. Seedling virulence frequencies for LR3A, 3BG, 3K8 and 23 returned to their 2019 levels. Virulence for LR24 was detected for the first time since 2017, and virulence for LR28 rose again for the 5th year in a row. We had this prevalent pathotype was detected in 2022 samples, and we identified 4 new pathotypes for brown rust in our 20. So that ends the brown rust. I'm now going to move on to the wheat and barley powdery mildew. So we do normally go through these in some detail, but today we're not going to feature too heavily on the powdery mildews, and the full results will be in the annual report later this year. So for the WPM, so that's wheat powdery mildew, these are the samples we received last year. We had 12 samples, they came from 6 counties. The majority of the samples did come from Cambridgeshire, but we also had samples from Scotland and Northern Ireland, and as you can see in Lincolnshire. They came from 12 varieties, so that's 12 samples, 12 different varieties, and those varieties included Cadivus cranium, Cadivus caelum, LG astronomer, Merit and RGT Rashid. So instead of going through all the virulence frequencies, I've just got some key bullet points. So I tested 19 single pustule isolates on the wheat mildew differential set, and 79% or more of the isolates carried virulence for PM2, PM3D, PM4B, PM5, PM6, PM8, MLD, MLTA2, MLTO and MLRO. Virulence for PM17, that one dropped from 100% in 2021 to 21% in 2022, but it's been previously seen at those levels before 2021, so I don't think that was anything of concern. Virulence for shamrock, that one also dropped from 50% in 2021 to just 5% in 2022 isolates, but again, that one's been seen at previously very low levels, so I don't think that's anything to worry about. No unusual outbreaks were reported, so it is thought that these changes did not translate into detrimental effects on varietal performance. And that's it for the wheat mildew. I'm going to now go on to the barley mildew. So for the barley mildew, we received 29 samples. They came from 7 counties. The majority of the samples did come from Northern Ireland, but we did have samples from Devon and North Yorkshire and Cambridgeshire counties. They came in from 19 varieties, and this time Cadivus Orwell was the most sampled variety. So my bullet points for the barley powdery mildew are that I tested 29 single pustule isolates on the barley mildew diff set. Virulence frequencies were actually really similar to 2021. 90% or more of the isolates carried virulence for MLH, MLRA, MLG, MLA6 and MLA12. Virulence for MLA13, that one rose to 34%, and it's not been seen in 2020 or 2021. No virulence was detected for MLO11 carried by Riviera for the 5th year in a row, and no virulence was detected for the additional cultivar. And looking at these results and comparing them with previous years, the population does seem fairly stable at the moment, despite the low number of isolates tested, especially in 2019 and 2020. So that's it for the barley powdery mildew. We can now go on to wheat stem rust. So this isn't a disease that I'm talk about very often, but you may be aware that it was seen at the end of the 2022 disease season, and you may have seen the reports on social media that stem rust was making a bit of a comeback. So Stem rust, this was the first sort of like big epidemic we've seen for stem rust since the 1950s. It has been picked up in recent years, but only on very isolated plants. But in 2022, we did have quite a few reports coming in from stem rust. Stem rust is not currently included in the survey, so we're not actually funded under the survey to do anything with stem rust, but we received 7 samples. So most of you may have sent your samples to JIC because Diane Saunders currently works for stem rust, but we received some here, and of course I was rather excited by stem rust because it's not something that I've been able to work with before. So I asked permission from Charlotte and Kostya Kanuka, who's head of pathology here at NIAB, if I could do something with these samples, and they agreed. NIAB would absorb the cost of that. And I bulked 5 of my 7 isolates in the growth rooms, and I've stored the spores as freeze-dried spores. And that's really important just because we will be using those isolates for future research projects. So it's great to have those in our collection here at NIAB. But of course, just bulking the spores wasn't quite enough for me, so I wanted to do something else with them. And I was given permission to do some seedling tests. And I took 2 single pustule isolates from just 1 sample and tested them on the RL and RL candidates. So just to look at the stem rust samples that I did receive. So I received 3 samples from North Yorkshire, from Gleam, Cadurus Guiam and Cadurus Renum. I received 1 sample from Lincolnshire, and that variety, I think it was an NL1 variety last year. I received some samples from Oxfordshire, but they were just labelled as various varieties, so I'm not quite sure what varieties they were, just a collection of leaves. We received 1 sample from Cadurus Webham in Cornwall and 1 sample from Swallow in Shropshire. And it was this one here from Gleam that I took forwards to my seedling tests, and no particular reason for that other than that it did do quite well in my growth rooms when I was bulking it up. I just thought I'd make life easier for myself and pick the one that was doing well, so I picked this one here. So my seedling test results. So I tested these 2 single pustules isolates of stem rust 22007 on the RL and RL candidates, and all varieties except Theodore were susceptible to both isolates at seedling stage. And I'd like to point out that limited pustule development was noted on RGT Wolverine, and I've got some pictures to show you next. But I just wanted to show you these pictures here first. So these are just a little snapshot of some of my seedling test photos, and it was really interesting that even on these seedlings that had only been, this was like at day 12 post inoculation, you can still see that stem rust was living up to its name and developing on the stem of these little seedlings. So this is Theodore. So this is on the 1st of December. So this is about day, I think this is day 14 by now. And you can see Theodore is resistant to both of those single pustule isolates. Yes, you could say that there's these tiny little pustules forming, and it has got some chlorotic spots. We have a scale for stem rust. It's a 0 to 4 scale. But Theodore, these pustules never developed any further than this, so we would class Theodore as resistant at seedling stage to these 2 stem rust isolates. RGT Wolverine, so slightly more stem rust was actually seen on Wolverine, and I've got the 2 pictures there of sort of the size of the pustules I was seeing, and to be fair, these pustules, they did develop further as the testing time period went on. So we have kind of a sort of like set testing time period, but because it was stem rust and I was more interested in these, I did leave these in the growth rooms for quite a long period of time after I'd officially finished scoring them. And these pustules did develop, but at the same, if you have a look at the picture in the oval, that shows what other varieties look like on the 1st of December. And you can see Wolverine is clearly more resistant than what than a lot of the other varieties. So we're not saying it's not as resistant as Theodore was, but it must be exhibiting some there, either sort of like intermediate reaction or partial resistance. And I just had a few more photos to show you. So it's you can see this characteristic shedding of the epidermis that was clearly seen even on these. So these are they're about 24 days old, these seedlings, and like I said, 14 days post inoculation. And it's important to note that stem rust pustules do grow larger on the underside of the leaf. So if you are looking at it in the late summer this year, always check the underside of the leaf. And I don't know if my video works, I just wanted to show you that. So this is the upper surface of a leaf. And then if you turn it over, you can see the stem rust pustules develop much more on the underside of the leaf. So just a quick stem rust summary. We had 7 samples were received in 2022. 2 single pustule isolates were tested from one sample, selected from one sample, and they were tested on the RL and RL candidates. Theodore classed as resistant at the seedling stage, and RGT Wolverine appeared to show limited pustule development. So for 2023, please send stem rust samples to Diane at John Innes, because she's still looking at stem rust. And more details, you can find that on the AHDB website. And I also wanted to point out the AHDB website has this really nice web page about stem rust. It has more information on the life cycle and the risk to UK wheat. And it also contains links to the stem rust presentations given by Diane Saunders at our 2022 UKCPVS stakeholder event, and the one she gave at the 2022 AHDB agronomist conference. And they're really informative presentations. So if you are interested in finding out more about stem rust, please do take a look at those. So I'd now like to pass you back to Charlotte, who will continue the rest of the presentation.
Thank you, Amelia. Okay, so now we're just going to quickly talk about sampling in 2023. So there's a slight change to the programme. This year we'll be focusing on the rust pathogens, so please continue to send us samples of yellow rust and brown rust, but we're not surveying powdery mildew this year, so please don't send us powdery mildew samples. Just an update, so have we seen any rusts? Yes, we have had some reports and we've had some samples in. We've had 3 yellow rust samples from Lincolnshire, so thank you very much for sending those in. And just to remind you that we welcome samples from any RL variety or RL candidate, and please send them from inoculated and preferably untreated trials. So sampling in 2023, just wanted to say thank you to everyone who sends us in samples. We know how busy the disease season can be, and we really do appreciate your time and effort in helping us with the survey. Every sample is important to us. As I said at the beginning, we try and provide each one that's sent in to us, regardless of the host variety or any other information. And we make every effort to give each sample the best chance of survival and aid successful spore multiplication. And we couldn't run such a successful survey without you, so thank you. Just to explain or maybe remind people how your samples are used, so the UKCPVS provides isolates for recommended list and variety list inoculated trials, and these are used to produce the disease ratings in the RL handbook. And the UKCPVS seedling data is now included in the RL lists to give another layer of information. The isolates or the information and isolates are also used by breeders to screen breeding lines. So this is really important for future commercial resistant varieties. AgChem and biocontrol research trial new products using the isolates, and also occasionally we have requests from other researchers. So the survey helps research projects, PhD studies, ongoing collaborations with other research institutes, really focusing on investigating the complexities of the pathogens at the molecular level. We have a good track record of participation in European projects. So without your samples, we wouldn't have such a good resource and valuable input into on the European scale. And as I mentioned, all isolates go into long-term storage for future research. So just because your sample isn't tested in that particular year doesn't mean that it won't be useful for future research. So just to show the sampling sheet, so please remember that when you do send in a sample to us, please fill in this sampling sheet and please fill in something in each one of the columns. As much information as possible, the more the better. And the website's on the top of the slide where you can find more information about sampling. Please place the leaf samples directly in the paper envelope. Please do not use a plastic bag. And send samples with just the words free post to UKCPVS if you're using our free post service. If you want to attach a stamp to send it via the first class or next day delivery, please send it to our Park Farm address, because this is where, so for those that don't know, NIAB has 2 sites in Cambridge, and the UKCPVS project is run from the Park Farm site. So please don't send it to the Huntington HQ site, as they can easily get missed or delayed. But again, more information is found on the NIAB website. So I just wanted to highlight a few of our take-home messages. So disease pressure was affected by a dry April and an extremely hot summer in 2022. So there were no reports of unexpected varietal breakdowns due to yellow rust. But what we're seeing is that the population remains diverse. But the good news is we're not detecting any virulence for the genes YR5, 8, 10, 15 or 24 in the isolates that we've screened. So if those resistance genes are in our breeding programs, that's really good news. Higher level than expected of brown rust reported on Theodore, and as Amelia explained, this is possibly due to virulence of LR24, and there's no unexpected outbreaks of wheat or barley powdery mildew in 2022. And I'm sure I don't need to remind you, but please refer to the AHDB RL handbook for disease ratings, and please do take the time to look at the young plant resistance in there as well as the adult, and remember to monitor crops very closely, and especially those varieties rated 8 or 9, and please send us in any samples. And as temperatures rise, watch out for brown rust and stem rust. So UKCPVS helps farmers make informed variety choices. It also helps to reduce on-farm inputs and reduce the spread of disease, and also helps breeders in the development of new varieties. And for further information, as has been mentioned during the presentation, there's the back, there's all of the old annual reports on the AHDB website. There's also the recommended lists and press releases on the AHDB website. Sampling information and postage and packaging information can be found on the NIAB website. And then also, if you're interested in a more global picture, there's also the Global Rust Reference Centre with a link on there that shows about what else is happening around the world. And this work would not be possible. It's not just down to me and Amelia. It involves a lot of people. So we'd like to acknowledge the funders, all the people that send us in samples, the team here at NIAB, so Janet, Helen, Anne, Megan, Edda, Sarah, and also our collaborator Diane Saunders and her team at the John Innes Centre. So if anybody has any questions, I guess we've got a short amount of time to answer those.
So we do have a few questions in the chat. And we're not able to unmute participants, so if you have a question, please write it in the question box and we'll get that through. So we've got a few questions about, the first one is, what methods do you use to revive and bulk out the rust isolates?
Yeah, so basically we first have to, when you send us a leaf sample in, I first have to put them in a little damp chamber, and they are incubated for overnight or a couple of days. And then when those pustules start to sporulate, I actually transfer them onto leaves of our susceptible varieties. And we have 2 rooms down in the growth rooms, one dedicated for yellow rust and one dedicated for brown rust. So basically, it's literally just leaf-to-leaf infection to bulk up those isolates, and then we can collect the spores off them, off those newly inoculated pots once they start to sporulate.
Okay, thank you. We've got another question for Amelia. How do you account for the high level of infection on KW6 stays in the off-season tests and the adult plant tests? Which do we believe?
Yeah, no, I did wonder that myself. So it was a little bit of a surprise to see that level of extays. I don't really know why that, because it didn't infect extays in its seedling tests, and it didn't infect extays out in the field. So I don't know if that's an environmental effect of being under those extended day lengths conditions. But yeah, but I'm afraid I don't have a clear answer for that. I would like, maybe if I could run those tests again, and we could have a look at that again. But yeah, so it's a surprise. I don't know why it infected extays, but it did. So.
Thank you. In the off-season trial, how did you stop the spread of rust in the airflow going around the greenhouse?
Yeah, so that's easy. That's why we actually selected 2 growth rooms. So I had 1 isolate, and these are spore-proof growth rooms. So that's why I had to separate those two isolates. So I had 2114 in one growth room and 21045 in the other growth room to keep them completely separate, so there's no cross-contamination between the isolates. And because they're spore-proof growth rooms, all that area goes through filters and HEPA filters, so they can't cross-infect between growth rooms.
And then one more I've got is, do you have isolates that don't carry virulence?
So if you go back in through the yellow rust collection, the older varieties have less avirulent on some of the differentials. So back in the day, then there was no virulence for maybe YR17 or 32 and things, or YR7. So yeah, the older collection isolates have avirulence for certain differentials, and of course even our modern isolates now have avirulence for things like YR5, 10, 15 and 24. So yeah, we certainly do have isolates that have avirulence.
We've just had 2 more come in. Hi, I'm curious on what method you use to assess percentage leaf area infected. Is it a visual estimate? So
for the off season, so yeah, if the there is actually an official key for assessing leaf area. It's in the AHDB inoculated trials protocols. But yeah, so, and for the adult plant trials, when we're assessing percentage plot area, there's an official assessment key for that as well.
Another one about the leaf rust nurseries. Was there any yellow rust present in the leaf rust nurseries that may have infected the levels of leaf rust?
Yes, indeed. We can't stop yellow rust coming in. And so this was my first year really of scoring the leaf rust trials, because normally we have one person doing the yellow rust and one person doing brown rust. But I was very, very careful with things like Skyfall and RGT Wolverine to carefully look for brown rust before the yellow rust got going. And in fact, with things like Skyfall, I had to put them down. The later assessments I did on those that variety, I couldn't record because the yellow rust was, it was unfair to score the yellow rust. So yeah, we are very conscious of the fact that yellow rust could contaminate our plots, but although you can't spray do a spray that will differentiate between the two rusts, but you just have to score your plots really carefully, and then when it becomes unscorable, you have to just make a note in the on the assessment sheet that it's unscorable.
Thank you. Another one's come in. Reports of yellow rust in X-stays already. Is it likely there are a few isolates X-stays are susceptible to but resistant to most?
Yeah, so we definitely find that X-stays seems to be susceptible to isolates. Some of them, I find that it's X-stays and Crusoe quite often go hand to hand, not all the time at seedling stage, but yes, there's definitely isolates that infect X-stays out there in the current population. I mean, so if you think about it, although we had 187 samples in, we only test 25 of those. So it could be that if I had tested more isolates or different isolates, then we would have picked up X-stays even in our seedling tests.
And then another question in relation to the question just before that about the leaf rust. Does that mean that the Skyfall leaf rust is divided by 2 and not 4 scores?
Yeah, in some case it was actually divided by 2 but not 4 scores. But this is the trouble when you have varieties that are highly susceptible to one or the other rusts. And you could argue, to be fair, the same with Crusoe and brown rust. Crusoe in the yellow rust trials also gets obliterated by brown rust later on in the season. But yeah, so but it's still an average. That's why you could see that some of the scores for Skyfall was so low that it didn't really even register as a 0.1% is our lowest, but the scores for Skyfall were actually below that because it's an average of the viable scores we had.
What about stem rust? What will be the movement of stem rust this year? Could it reach up into Scotland?
So if you have a look at Diane's talk, it does actually talk about quite a lot about the movement of stem rust and how it could be. So last year we had Saharan winds coming up from the southern Europe and that lot, and that may have brought the stem rust across. We also had the high temperatures, but yeah, so spores can travel for thousands of miles up in the atmosphere. So yeah, it is highly possible that you may see it up in Scotland, especially if you get nice weather up in Scotland. Because it does like the warmer temperatures.
That's all the questions we've got at the moment.
Okay, thank you very much, everyone.
Thank you.
Right, well, welcome back, everybody. It's now a pleasure to welcome Lisa Jorgensen from the Department of Agroecology Crop Health at Aarhus University, or Aarhus, I should say, perhaps, University in Denmark. Lisa's got a very wide experience in disease management in cereals, including variety resistance, fungicides and integrated control. And of course, she does bring a very useful European perspective to what's going on with our plant pathogens, and played a big part, for instance, in the Rust Watch project, which I think concluded last year, actually, but obviously the results are still very relevant. So over to Lisa. Thank you.
Thank you very much for the word and the opportunity to share some input about IPM in winter wheat with you, focusing on winter wheat diseases. The slide you see here is these 8 IPM principles. I know Brexit has happened and you're not part of the EU family anymore, but I'm sure that these still play a major role. So it's all about how we can prevent attack. Nothing happens now. You are hidden webcam. So I shouldn't share my webcam. So, okay. So, switch this on. Is this better? I hope. Yeah. Everything got very quiet.
Hiya, we can see your slide.
You can't see my slides.
Yes, we can.
Yes, but they don't move forward when I file them.
Yeah, just carry on.
But
Excellent.
they
Yeah, we
don't.
can see you on your slide.
But they don't go forward. So there's something. They are stuck somehow. So what to do there?
Lisa, are you clicking on your PowerPoint?
Yeah, I'm clicking. No, yes, I'm trying to get it to move forward. Yes, as it should do. I think. Do we have to just make a new entrance into sharing it?
Yeah, maybe try that.
So you see it now, I hope.
Yep, the circles
Yes.
are moving forwards now.
Sorry for this. Well, the 8 IPM principles, which is, I think, familiar to most, it's about preventing attack of diseases. It's about monitoring and threshold use. It's about using as little as possible of the traditional chemistry, alternatives if possible, specific fungicides, and keep fungicide input as low as possible. And when it comes to resistance, it's all about diversifying the choice of chemicals you use and making sure that you don't push fungicide resistance unnecessarily. So these are sort of the basic principles, but how they are going to be taken into account in the individual countries is very open at the moment. But I'll share with you some of the elements which have been included and in the Danish perspective. So less dependent on chemicals, more cultural method, and so. I followed in the morning all your good data on the resistance characterizing from the recommended list. We have similar in Denmark. Also, we see big benefits from the breeders bringing varieties forwards which show good resistance. This is an example of Septoria resistance. We can see that we actually had available varieties with a good resistance, which are also the high yielding ones and are also the ones which the growers are actually choosing. We have specifically had a lot of discussion about variety mixtures, cultivar mixtures, and so the good question could be, why should we grow these mixtures? So typically, when we compare a mixture, the benefit of a mixture, we always take the average of the 3 components, and then we compare that with the output from the mixture to see if there's a benefit or a negative benefit or input from this mixture. So when I show data in the next slides, it will all be about this comparison between the data from the mixture and the average of the components. So the concept is that they should give less diseases and slightly higher yields in mixtures have also been seen. And that should bring us to a situation where we have less need for fungicide input. Another element of the concept is that we should have less disruption. We should disrupt selection, and that is with respect both to fungicide resistance, but also for virulence. Make sure that we don't have, we can minimize the aggressiveness of races developing. Other benefits could also be that the mixtures are more tolerant to variable climate changes or climates in general. It could be winter hardiness, where they could compensate if one was more sensitive to winter hardiness. Or it could also be that there was differences in drought tolerance, for instance. We've also seen that these varieties, the mixtures can be less likely to lodge because they support each other. So the need for input of plant growth regulators is also something which could be reduced. So the two figures to the right just indicate that over time we actually see this, that the variety gets more susceptible, and also with the increasing use of fungicides, we can see that they drop in efficacy over time. So the varieties mixtures could not solve all these problems, but they could help to slow down the process. In 2023, the area sown in winter wheat in Denmark, 39% of the area is sown with variety mixture. And as you can see from the data here, there's been a steep increase from 2020, 5%, in 21, 11, 22, 25%, and this year, 39%. So a really major uptake of this growing variety mixture in the farming community. And the other varieties which are grown to a major extent is those which are also having what we see good resistance in general. So farmers really try to actually grow varieties with good resistance, and variety mixture is part of that equation. The benefits from growing the mixtures, I mean, we have studied in various projects, and we have particularly a PhD study done by Rose Christoffersen, and there we can see over time that the benefit on Septoria triticale blotch has been an overall benefit of 14% reduction in disease in the mixture compared to the components in the mixture. And the more, if we include varieties with more susceptibility, then the benefit is even bigger. It's also an indication that 4 cultivars is better than 2 cultivars in the mixture, so also that is playing a role. In the Rustwatch project, which we had together across the whole of Europe, and UK was also part of this and also part of delivering these data, which I'm showing here, that we had 16 trials with yellow rust, and there we had a 30% reduction in attack of yellow rust in the mixture compared to the average of the 3 components in the mixture. And this was, the mixture was here including a susceptible variety, a slow ruster, and a resistant variety, but clearly a benefit. And also when it came to brown rust, we had less data, but 6 trials did show that we had an 11% reduction in attack. I mean, it's not rocket science that we see these reductions. There's been previously historically investigation also showing benefit both on mildew and rust diseases from mixtures. But often the take up in the farming community has not been significant. If we look at the yield component, we can see that there has been a yield benefit in the mixtures compared to growing the solo varieties. And this benefit is particularly high or the best if we have the untreated plots. The more fungicide treatment, if we have 3 treatments, we are less than 1% yield benefit. But if we have untreated, we have more than 4% yield benefit. So if we should put that into perspective, then if you just plaster your crop with fungicides, well, then you won't see a benefit. When we come to the disruptive selection from the mixtures, we have actually been investigating the selection for specific CYP51 mutations. And there we saw that some of the well-known CYP51 mutations were less developing in speed in the mixture compared to the average of the varieties in the component. So this also was positive that we could actually see this reduction. When it comes to the virulence, to show that there is a benefit on virulence against rust or septoria and other diseases, there's not that much data available, but there are some French studies coming up which have shown that particularly against SDB6, the virulence against SDB6 has been reduced when growing variety mixture. So that is also looking positively that there could be a benefit here. The reason why the Danish farmers have picked up this, I think one of the main elements is that stakeholders have agreed on this is a good idea. And we have this platform which is called Sort Info. And if we look last autumn on the 10 most best yielding varieties, we could see that 3 of those mixture were actually, or 3 of the 10 best ones were mixtures. So the fact that you could show easily to the farmer that these mixtures are actually behaving well, both when it comes to disease levels, but also to yields, has had a significant impact on the uptake of growing cultivar mixtures. There is also a yield benefit, which is significant, I think, that both that you have a benefit in yield. If we take all data from the last 10 years, we have had a 2.1 deciton better yield. And there is an extra cost of the seeds, which has to be deducted. But if you can save one fungicide treatment, which is typically what we advocate, that instead of spraying 2 to 3 times, you can spray 1 to 2 times, then you have a saving there. And overall, there is an economical benefit. It's not that it will solve all problems, but it can give a stability and maybe ensure against major failures, as we saw with benchmark and yellow rust for some years ago. It's a good option for wheat and barley, which we feed to the animals, whereas it's of course more problematic when we talk about bread wheat and also malting barley. Bread wheat, there might be an open door coming and could be used there, but in the malting barley industry at the moment, there is quite a reluctance. Another IPM element in taking the prevention is, of course, time of sowing. And this is just illustrated here, where we have combined nitrogen levels with time of sowing, early sowing or late sowing. And we see that septoria is very influenced by the time of sowing. That if we sow too early, then we get more septoria. We also get more take-all. We also get more barley yellow dwarf virus, and all that will also increase the need for, or the reliance on pesticides in general. So we advocate that the farmers should try to delay the sowing. We have an issue here because there's another political driver here which is encouraging the farmer to sow early, because then the nitrogen take of the crop is better, and then they don't have to have such a big area with set aside. So it's a good example of political activities going in two opposite directions. The use of thresholds and decision support system is, of course, extremely important. And when we look here, just taking the Danish data on the yield benefits in the individual years from the use of fungicides in wheat, we can see that we have years like 2008 and 18, where there has been absolutely no benefit from spraying with fungicides. So those years, the farmer should not have sprayed. Most years, of course, it's true to say that there is a benefit from spraying, particularly with the green line showing the grain price, which we currently have. So there need to be a consideration of tools to help to guide you on what you need to do. So this red button here kind of should be the one giving you the magic answer, do I need to spray or not? And I'm sure many of you will know that it's not such an easy game to actually make the decision on whether you need to spray or not. At the end of the season, we are all very clever and know whether it was the right decision or not. But it's also true to say that if we monitor the crop, if we follow the weather data, then we do have some indication on how intensely we need to spray our crops. And typically farmers have their sort of spraying schedules. Is it a low risk scenario where you will spray maybe only once, which if you have a variety mixture or a good resistant variety, or if you have a susceptible variety, then you might need to spray 3 to 4 times. So it's important that you know from the beginning where you are, that you have a low or high risk, and the variety is of course a very important element in that equation. This slide here shows that decision support systems can actually be a help. We have the system Crop Protection Online, where we have been validating it for many years in trials, both with very susceptible and less susceptible varieties. And when we look at the output, it's not that it gives better disease control or better yields. but it gives a variable input depending on the need. And overall, we could save 37% of the fungicides compared to a 2-spray strategy if we had implemented this decision support system where we looked in the crop and that we followed the risk assessment. And particularly again, Septoria, we know that humidity drives the disease, and there relatively simple rules has been applied that you spray against Septoria if you have had 4 days of precipitation, or in a resistant variety 5 days of precipitation starting at a later stage. So these are rules of thumb, which I think that most farmers they recognize today, and also to at least some extent apply before they treat their crops. We have a new model, the humidity model, which is also being implemented, and that's recommend treatments if we have had a very, very wet day, more than 20 hours with continuous high humidity above 85%. So there's actually data also showing that that model is giving answers or guarantees of proper recommendation in comparison also with crop protection online. So just one word on this. I mean, there's a new decision support platform. IPM Decision is an EU project where there will be a platform trying to implement the various available decision support systems to make it more easy for the farmers to access the different decision support systems. This is actually led by Neil Pavley from ADAS, and should be launched, an upgrade of the platform will be launched this spring. So keep an eye on that. Fungicides are available, and we are still, despite all the IPM, still relying a lot on these chemicals. We have different groups. Won't go much into details here. You have it all in UK. All the red ones listed here we don't have in Denmark, so our arsenal of products is much lower than the one you have available. However, we need to apply the products which are most effective, and the optimization of the dose is an important element. I mean, for the Danish situation, and also when there was a survey across Europe, it came out that all countries had some degree of reduced rates being recommended, and the optimal dose is of course a variable size depending on the season. And the figure to the left is one taken from AHDB's own work, whereas the one to the right is just Danish data indicating that if you have a very severe level of Septoria, then the input should be with a treatment frequency between 1 and 1.5, whereas if you have a very low disease pressure and a resistant cultivar, well, then you need most likely less than 1 treatment frequency per season. So these are based on trial data which we have analyzed. John Lucas, in his introduction, just touched very briefly on biological control of diseases, and this is something which is highlighted a lot, that there should be a serious look into alternatives to the current chemical fungicides and pesticides in general, and we have also been looking into this. Everything seemed to work well in a petri dish, maybe also in a pot trial, but when the further you get out in the field, the more challenges you have to actually get the good responses from some of these biological control agents. In 2022, we carried out some field trials in Denmark where we looked at all the alternatives which we could sort of be aware of. They included products which was bacteria, there was fungi, there was phosphonate, there was some which were, a lot of them based on where sulfur products was part of it. And what it came out was actually that all the sulfur products did do quite a decent job when it came to control of Septoria, in line with what we could see from 2 half rates of proline. Sort of, we know it's a nasal, at the end it's also worn out a little bit because of resistance. So we had a performance in line with that. When it came to yield, I mean, the picture was less good because the persistence of the alternative chemistries were not so good. And compared to the top, which is sort of a traditional chemical control, most of the treatments did not really provide a significant yield benefit. So there are some things which can be used, but there's still a long way sort of to see that they can take over from the efficacy which we recognize today from the chemistry. We also, in the Rust Watch project, actually had some trials where we were looking at alternatives, and when it came to rust, it seems that we are even more struggling to find alternatives to the traditional chemistry. The picture to the left is from our field trials area, where the right is showing some nice green fields where we have been giving chemical control of yellow rust, and the yellow patches there are untreated, whereas the next block is where we test some biological substances, and it's much more yellow, as you can see, and there are some green spots there, and that's the chemical reference which shows up. So kind of an opposite picture to what we see at the right. So we have actually, there are challenges there. We need to search for new alternatives if they are to be replaced against rust diseases. I won't touch much on fungicide resistance because Nicholas will be doing that. But we do see, sorry to say this, we do see Ireland and UK, the British Islands, as a hotspot area for developing of fungicide resistance. We know if we take Septoria that you have severe attack of Septoria, and maybe also more than, at least in some areas on the continent. But there are also, which I think is a tendency to apply more frequently some of the actives than we see in the continental Europe. To the left, we have some of the findings of SDHI mutations, and there it has been clear that we have, in France and Germany, they have been recommending only one SDHI treatment per season, and similar in Denmark, whereas I think in UK and Ireland, there have been a higher level, more has been recommended. So this is, of course, of concern that we have these developments going on. And when we think about anti-resistant strategies from a Danish perspective, we really like to see that the single active should only be used once per season in order to keep down selection pressure and keep the products alive for longer. And then, before coming to my last slides, this is also something, I think, a message which is important to take home, and it's actually how clean should the field be before you should start to get worried. I mean, there's 4 pictures showing different degrees of Septoria, and I mean, we should not be too worried if there's a little bit of Septoria left, same for mildew and other diseases down on the bottom. It's not the end of the world. And I love this statement made by John Lucas that we should not be too keen to be clean. And I think that is also important, that we should make sure that we don't overdo the job. And our long history with use of reduced rate has actually helped us, I think, to also keep down the selection, because we have not been overdoing the dose when we have been applying things. So, just a short summing up. So factors which will lead to more IPM, they're very obvious, and it's not like it's new rocket science. Grow resistant cultivars, and from our perspective, cultivar mixture seems also to be a good option. It's important, I think, that the farmers learn to delay sowing and adjust the sowing time to keep down the decision pressure. We should focus more on the use of decision support system, and keep on only applying fungicides, effective fungicides, and the appropriate rates. Critically thinking what is the economical optimum when we look at the dose rate. We should ensure good anti-resistant strategies. And then when it comes to alternative chemistries, that might be, I think, valuable in the future, but right now it's a little bit traumatic maybe to think that they could just take over what we know from the good chemistries. So with that, I will finish. I've just showed to the right this graph of the kilo actives used in different parts of the world, and you can see that there are substantial differences between countries, but there's also substantial differences in the crops which are grown, which impact a lot, of course, the need and the need of specific input you have in different countries. But there are major differences. Thank you.
Thank you very much, Lisa. We're a little bit over time here. I think there's one question I saw, which was about reducing the dose of fungicides might increase the chances of development of tolerance to these fungicides. I don't think there's a conclusive answer to that, to be honest, because studies have shown varied results when you change the dose. I don't know if Lisa's got a quick comment on it.
Well, it's been a discussion as long as I have been in this game, and I think overall there are several studies indicating that you don't actually have a significant increase in resistance when you reduce the rate. More opposite, I think, because the more part of this, don't be too keen to be clean, because if you really bang it all back, then you are more likely to select for some which are more tough and more resistant. So I think for the scenario we have had and a long history of using reduced rates, we have not been seeing more resistance than anywhere else.
I think there's some quite good publications on this from Frank van den Bosch's group and Neil Pavey
Sure.
and co. So,
Sure.
but it is quite a complex debate.
Yes, I agree.
Okay, I think we ought to finish there, Lisa. Thank you very much indeed for your contribution. Sorry we overran a bit, but you had some technical troubles at the start. So I think we ought to move on. Many thanks. Okay, so hopefully a seamless process to our next speaker, who is Nicola Hawkins. Nicola is a postdoctoral researcher in plant pathology at NIAB, and actually she holds currently a prestigious BBSRC Discovery Fellowship researching the evolution of fungicide resistance in plant pathogens. So over to Nicola.
Good morning. So I'm going to be talking about the evolutionary biology of the pathogens, and there are two main reasons why that matters. Two main reasons why this matters in the field. So one of those is that these pathogens can overcome host resistance, and the other is that they can evolve resistance against chemicals, so resistance to fungicides. But in fact, whatever control measure you try to use, that will be selecting to favour any strains that can overcome that control measure. So any control measure is at risk, but that doesn't mean that all control measures are at equal risk. So the actual risk for any given measure, well, it will depend on how complex a step is needed to evolve from sensitive to resistant, and it will depend how much of that genetic variation is there. So that could be variation already there in the population, or it could be the mutation rates generating new variants, and it depends on how strong is the selection. So the complexity of the evolutionary step, this tends to depend mostly on the particular control measure. The genetic variation depends mostly on the pathogen, and the strength of the selection is what can be altered by the management. So to explain what I mean by how complex an evolutionary step, well, in the case of fungicides, if you just got one fungicide with a single target site, then often it's a very simple step from sensitive to resistant. So for the strobilurins, that would just be one single mutation to go from sensitive two very resistant. For the azoles and the STHIs, it's slightly more complex, so one mutation might give a partial shift, but then once a few mutations accumulate, then you have higher levels of resistance. If you then instead use a mixture of fungicides, then a slightly more complex change is needed, so you would need changes in those different target sites. Although if you're using a mixture and there's already resistance to one of those mixing partners, then you're back to the situation of only needing one mutation to be resistant to both. And then the lowest risk would be the multi-site fungicides. So you would need either several target sites changes at once or non-target site mechanisms to give resistance to those. Now, of course, there are other reasons why we don't just use multi-site fungicides. So the single sites are more systemic, they're more curative, and with the multi-sites being so broad, there are more concerns about off-target effects, and that's why we've lost chlorothalonil. But just in terms of resistance risk, that risk would be lower. And we see a similar situation really in terms of the varietal resistance. So if you've just got one major resistance gene, then that's easier for a pathogen to overcome compared to if you start stacking different genes or even introducing quantitative resistance genes into the mix as well as major genes. And then perhaps the lowest risk of all would be non-host resistance, and there are some researchers looking at using that as a more durable way to have resistant plants. In terms of the genetic variation, here we can see the life cycle of Septoria, and we can see that it talks about the pycnidia. So these are the clonal spores where you get multiple cycles all through the growing season, building up to really quite a high population in a bad epidemic year. And then at the end, we see those ascospores, and that's the sexual cycle. So there we're reshuffling the genes into new combinations. So you've got this new variation from reshuffling the genes once a year with the ascospores, and then we've also got this polycyclic disease with all these multiple clonal cycles building up to large populations. And so this combination means there is a lot of genetic variation. So this makes it high risk, and there's not so much we can do about those risk factors. But the strength of the selection is the part where management can make a difference. So really, selection is stronger if you're really relying on one single control measure. If you're going from a really high disease pressure and wanting to go from heavy infestation to completely clean with just one measure on its own, then that's really strong selection. That's pretty much a recipe for resistance. So resistance management, we would look at either using less of that single measure, so that could be reducing the number of sprays of one product, it could be adjusting the dose rate, and we could look at combining with more different measures. So this could be mixing or alternating different fungicides, it can mean growing a resistant variety as well, and it can mean cultural control measures such as the crop rotations or management of stubborn volunteers. And it can also mean getting the spray timing right, so spraying in time rather than once the epidemic's already out of control and then trying to clean up afterwards. And so these three factors will give us the combined risk level. So we have the risk that comes from the fungicide classes in terms of how easy it is for resistance to evolve. We have the risk from the pathogen, so how variable it is, and some other factors involving the pathogen genetics. But then we have the agronomic risk, and these three factors multiply together. So that means if you have high-risk fungicides being used against a high-risk pathogen, then it's especially important to consider the resistance management to bring down the agronomic risk as a way to reduce that total. So that would mean for a high-risk pathogen with a high-risk fungicide, that makes it, we have to be a bit stricter with the resistance management guidelines. So for example, you might be told with a high-risk fungicide, only use it in mixtures, only use 1 spray per season. But to tailor the resistance management guidelines to the risk level, that means we need to have a resistance risk assessment for new fungicides before we have resistance. And so some of the research I'm doing at the moment is looking at how we can predict resistance in the lab. And so we have populations, and I'm working mostly with Septoria of a fungus in the lab. We use UV, so we have lots of mutations there. We select with the fungicide, and then if we get some lines where it can grow at higher concentrations, we take those lines forward and carry on selecting. And then at the end of 6 weeks, we look at those mutants and we'll test their fungicide sensitivity, and we'll also do the genetic analysis to see which mutations we're getting. So a plate might look something like this. So from the bottom row to the top row, we have increasing doses of the fungicide, and then the 6 columns have different lines, and you can see here lines 1 to 4, they're still sensitive so far, so they're only growing up to row C, but then in the last 2 columns, they're growing all the way, so these are some resistant mutants. And in this case, these mutants are resistant to carbendazim. So you might remember the benzimidazoles from previous years, and that's a deliberate choice to use fungicides where we already have the benefit of hindsight, so we can compare what's happening in the lab to what's already happened, what's already finished evolving in the field, to see if our predictions are accurate. And then if we're able to test these predictions and to predict more accurately, this should improve our predictions for future fungicide classes in terms of what is the risk level and in terms of what particular mutations we need to be looking out for. So now I'm just going to move on to some of our data from monitoring resistance in the field. And again, this is in Septoria. So this is Prothioidestheo, which we use in the lab for testing prothioconazole resistance. And going back to 2003, we had a relatively sensitive population, and then by 2013 there's been quite a big shift, and by 2020 another big shift again. From the 2022 data compared to 2021, what we're not seeing is new, even higher resistant mutations at the upper end, but what we are seeing is a bit of a shift in the middle of the population. So it's all within the range we've seen before, but within that range, we're seeing slightly more of the isolates that are slightly above the previous average. So it's not big jumps, but there is a continuing slide, as we've seen for azoles before. We look at the mefentrifluconazole, so for example, that's the Revysol. We can see it's not been around for so many years, or within that time. In 2019, there were some more highly sensitive isolates, and we've lost those, but the bulk of the population is really still quite similar so far. So if we keep up the resistance management, hopefully this will be useful for a while yet. And this is looking at the cross resistance between those 2 azoles from the website in Hampshire. So if we had complete cross resistance, we would expect this to be one straight line. If there was no cross resistance, we'd expect a complete scatter. And we can see in this case, the association is quite loose. There's quite a bit of scatter, but they're not completely unrelated. So we would still be saying always mix either of these with a non-azole rather than with each other. But if you're using an azole at different spray timings, then it might be worth using different azoles at each of those points. So moving on to the SDHIs, here's some data for bixafen from different sites, and we can see the bulk of the population is fairly similar, but there are a few extreme isolates coming up at some sites. So I'll keep monitoring those. And if we look at cross-resistance with some of the new SDHIs that should be coming online in the next couple of years, again, it's not a perfectly straight line, but if you look in the upper right-hand corner, there are some isolates cross-resistant to both. So these are still one mode of action. They need to be combined with different modes of action, although the absolute numbers are lower for some of these new compounds. So they're cross-resistant, but you can expect some higher activity. And again, with these new SDHIs, hopefully with the right resistance management, it will keep the class as a whole useful for a bit longer yet. Finally, looking at the QII, we can see perhaps some variation in some of these sites, but so far nothing that would really be ringing alarm bells in terms of serious resistance mutations coming up. So again, good news so far, but keep up the resistance management. We don't have resistance yet, but they are considered one of the high risk groups for that to evolve at some point in the future. So to summarise that situation, with the azoles, we're continuing to see a slide in sensitivity rather than any big jumps, and some partial cross resistance. Similarly with the SDHIs, we're seeing mostly these medium sensitivity isolates, but at some sites perhaps a few more of the resistant ones, so we do need to keep monitoring those in case the frequency increases a bit further. And the new SDHIs in the pipeline are showing good activity so far, but again we need to keep monitoring these more resistant mutations. And with the QIIs, no resistance so far, but we want to keep it that way as long as possible, please. And also with some of the research that I'm doing in the lab now, hopefully we can improve resistance risk assessments for future products. So for further information, if you look on the AHDB website, one of the key things you'll find here is the fungicide performance dose response curves. So the sensitivity data I've presented is testing in the lab, but if you want to see how that actually relates to control levels in the field at field doses, then you can look up those curves from the fungicide performance trials. And also there are some resources from FRAG UK, so the Fungicide Resistance Action Group, including guides for fungicide resistance management. And I think there'll be a new edition later this year of that management guide. And another place to look is FRAC, so this is the Fungicide Resistance Action Committee, and that's an international group of the AgChem companies. And there you'll find a lot of more technical information in terms of the different fungicide classes, but also some videos explaining the basis of resistance and the different modes of action. So that could be worth a look if you're interested to find out more about the resistance. So just to acknowledge BBSRC, who are funding my evolutionary biology research, and AHDB, who've supported the field monitoring. My colleagues at NIAB, Pooja, the senior technician, who's running the sensitivity assays in the lab now, and Kostya, who's leading the plant pathology work. And also my former colleague Bart, who's now at Wageningen, but led the fungicide work at Rothamsted and then NIAB over many years.
Thank you very much, Nicola. It's a very clear presentation. Thank you. Has Catherine picked up any questions?
No, none have come through so far.
Just seeing as we're trying to catch up time, Nicola, just a comment. I think that Lisa's presentation had some interesting aspects in terms of combinations of fungicides, reduced fungicide dose and cultivar resistance. Do you have any comments on that approach? I mean, we did have a question about whether the reducing the dose is in some ways potentially a risk, but I think there's been some quite good publications recently on combinations of more resistant, not like very resistant cultivars, but if you have ones that are sort of an intermediate resistance, it still gives a benefit.
So yes, so looking at the effective dose rate is one of the areas where better predictions of what sort of mutations to expect can really inform the management guidelines. So for example, it's where we see a big difference in the advice between herbicides and fungicides. So with herbicides, there's a lot of metabolic resistance, a lot of quantitative resistance. So if you have a low dose, the first quantitative step is enough to be selected for, and then it kind of gives those stepping stones to go up gradually from sensitive to resistant. Whereas if you go in with a high dose, you'd need that really complex evolutionary change all at once. With fungicides, because a lot of the time we're seeing these target site mutations, so it can already take that big step all in one go. A higher dose won't necessarily stop that, and it's just giving stronger selection. But again, this is where it's not necessarily one size fits all for resistance management, and it will really be useful to know what sort of resistance we're anticipating and really tailor the management guidelines to the fungicide class and to the resistance mutations before that resistance happens.
Thank you very much. Okay, we better move on. So our next talk is from Paul Nicholson from the John Innes Centre. I hope Paul's out there. There he is. So just to introduce Paul, he's a group leader at the John Innes Centre, and I've known his work over a long period. At the moment, he's going to talk about Fusarium, but he has actually spent a long time working on the genetic basis of resistance to a number of pathogens of wheat and barley. So over to Paul.
Thank you, John. Whoops. Apologies here. Oh, that's because of. Oh, where have I gone? Sorry. Here we go. Does that show my full screen now? Hope so. It's good afternoon, everyone. So I'm afraid I'm a poor replacement for Diane Saunders, who's jetting around the world, and so you're getting a bit of Fusarium as a distraction from septorias and rusts. So we're looking at both resistance and susceptibility to this disease, because we think that there may be two different ways of approaching control. So for those of you who are not familiar with Fusarium head blight in the UK, it really became an issue post 1998, when we saw the first major epidemic in the UK. And you can see here the symptoms on wheat on the left and barley on the right. And if you go into the grain, the infected grain tend to take on this powdery pinkish discoloration, and they're usually extremely light because of the fungal colonization of the grain and consumption of the carbohydrate and starch. The pre-1998 UK population causing head blight was generally made up of Fusarium culmorum and another species formerly called Fusarium nivale, now it's called Microdocium nivale. Post 1998, we started to see a new pathogen, and this was Fusarium graminiorum. And this pathogen is of major concern to us because of its production of an airborne inoculum. Calmorum is a splash dispersed organism with no aerial dispersal phase, whereas graminiorum is homothallic, which means it doesn't require finding a mate in order to produce the airborne spores. It can do it all on its own. And we believe that these issues arose because of the growth of maize in the UK, starting in the southwest and now spread right across to East Anglia. On maize stubble, Fusarium graminiorum undergoes this sexual reproduction, and that releases ascospores which can infect subsequent crops. And one of the major issues with Fusarium head blight is do not ever sow wheat after maize. So maize wheat rotations, as the Americans learned to their cost, is an extremely foolish way to go. So avoid wheat maize rotations. And in addition, because of the ascospore production on stubble and detritus, avoid minimum tillage as well. So there are a number of challenges to improving Fusarium resistance, and in the UK our varieties tend to be extremely susceptible. These issues arise because of the interactions between environment and host susceptibility. Resistance can only be assessed in adult plants at flowering. It's a head blight after all. But even here, the susceptibility varies throughout the flowering period. And so it's essential that inoculations are all carried out, if you're making comparisons between two lines, that they're both compared at the same developmental stage, mid-anthesis. The genetics of resistance are generally complex, and so resistance tends to be quantitative. There are no silver bullets, no major gene resistances that are really available to us, and no variety exhibits complete resistance. So as disease pressure increases, so even the best varieties will collapse under that pressure. And as I've said, resistance is affected by environment, such that if you're carrying out your disease assessments under low disease pressure, most things will look resistant. High disease pressure, everything will appear susceptible. So it's extremely difficult to identify lines which have got truly useful levels of resistance. And all of this then leads to breeders and scientists requiring multiple testing across years and different environments. And we've got to consider that it's not just the yield loss which is critical, it's the accumulation of mycotoxins in the grain, and in particular deoxynivalenol or DON, which is a controlled substance which leads to the rejection of loads if quantities go above a certain threshold. For those of us working on the genetics, it becomes really complex because of the huge genome size of wheat and the fact that within wheat there are 3 independent genomes all segregating with their various characteristics. DON, we still really understand relatively little about why the fungus produces it. It's a mycotoxin. It's very toxic to human and animal consumers, particularly pigs and chickens, but it's also a phytotoxin. And this means that in wheat, it acts as a virulence factor, and it's the agent which allows the fungus to move within the heads. So following infection, the fungus moves, and it's the DON which allows that movement. It's not a virulence factor in barley. So if we look at resistance, we've got following infection by the fungus at a particular point on the head, the fungus tends to move down the spike and the mycotoxin symptoms develop above the spike. In barley, the fungus is restricted to the spike that it infects. It's unable to move within the spike. And this is so-called type 2 resistance. And one of the mechanisms involved in this resistance is barley's ability to detoxify DON. And the DON is modified by the host, the barley plant, transforming it into a glucose moiety added to the DON, which makes this then non-toxic to the plant. So this allows barley to restrict the movement of the fungus, and with that reduce the levels of mycotoxin accumulation and damage. So this is one of the mechanisms that we have, that we know of in barley. Using a GM approach, genes from barley have been transformed into wheat, and it's been shown that those genes also function in wheat to enhance resistance and reduce mycotoxin accumulation. So if one doesn't want a GM approach, the challenge then is to look for wheat varieties that contain versions of these glucosyl transferases, which are capable of undertaking this transformation of DON with greater efficiency, so to reduce the effects of DON on the plant and reduce DON accumulation. If we're looking for sources of resistance, most of the resistance available to us is coming out of Asia. There's a little in Europe and South America, but most is coming from Asia. And we consider resistance as being of two types. Resistance to the initial infection of the heads is governed by what we call type 1 resistance, and the spread within the head is the type 2. And this resistance, particularly the type 2, has been identified in a number of varieties, and here it does have a major impact on the susceptibility. And so a variety originating from China, Sumai 3, has a very potent resistance on the short arm of chromosome 3b, which is termed FHB1. And this has been studied for well over 20 years. And only now are we beginning to understand what that gene may be and what it might do. In 2016, there was a publication which announced that the gene had been cloned, FHB1 had been identified as being this chimeric lectin with agglutinin domains, whatever that might be. Unfortunately, a couple of years later came a publication which demonstrated that this was not the resistance gene. The gene involved seems to be this histidine-rich calcium binding protein, which is sitting next door. And yet, even here, there was controversy over the nature of the resistance. Was it a major gain of resistance, or was it the loss of a function, the so-called loss of a susceptibility factor? And really the research is still ongoing to try to differentiate between these aspects, and even to establish whether we actually still have not found the right gene. And personally, I believe we may still have identified the wrong gene, but that's something for another time. But it just demonstrates how it's taken researchers across the globe about a couple of decades now in order to go from knowing about a gene to actually identifying that gene and being able to give the breeders tools with which they can handle that resistance. I'm just going to skip through this one in the next because of time and describe another aspect of Fusarium head blight, which is where we're focusing our efforts. Most people are attempting to introduce resistance into varieties in order to enhance resistance. But what was recognized a while ago, back in 2006, was that actually you can increase resistance by removing bits of wheat chromosome. And this diagram here identifies the wheat genome, in which there are 7 groups with 3 genomes in each, the A, B, and the D. And what it shows is that those chromosome arms in green, removing those chromosome arms leads to an increase in resistance. And so, an increase in susceptibility. So this is what we would normally regard as being a resistance factor present. But on some of the arms, and in particular we'd focus in on 4D here, removing that chromosome arm actually leads to an increase in resistance. And so we believe that there is some factor present on that arm which is conferring susceptibility. So our challenge is to identify it and remove it so that breeders can use those varieties that are identical to what they currently have and yet lack this susceptibility. And because of this mechanism, it would leave these materials as ideal candidates for gene editing. So with the potential changes in legislation, it would give breeders an entirely new toolkit to approach increasing Fusarium resistance through elimination of susceptibility factors through gene editing. We've been focusing on 4D, and we showed that it's restricted to a portion of the 4D chromosome, and currently we're working on that resistance, and we've shown that small deletions can lead to very marked increases in resistance. So we have Powdery, the susceptible line, a line carrying a deletion on chromosome 4D showing greatly increased resistance. And we're looking at additional deletion lines, and it's allowed us to narrow down our search to around 30 genes, which we believe one of which is responsible for this susceptibility. So once we've identified that gene, it gives breeders a target such that they can eliminate that either through conventional breeding or through a CRISPR-based approach to enhance Fusarium resistance. So if we're going to increase resistance overall and eliminate mycotoxin accumulation in wheat, we've got different complementary approaches. It's to reduce susceptibility by increasing resistance factors like FHB1. We can enhance the detoxification mechanisms in wheat through identification of glucosyltransferase transferase activity, so as to reduce the mycotoxin accumulation and its potency. Factors I've not mentioned, but also for breeders, increasing height and anther extrusion, as anthers are involved in susceptibility to Fusarium head blight. And then finally, we have the approach of eliminating susceptibility factors. And it's going to be through a combination of these approaches and the stacking of these characteristics within an ideal variety that should ultimately lead to the reduction and potential elimination of FHB and associated mycotoxin accumulation as a threat to consumers. And so with that, I would just thank my colleagues, both present and previously, other members of the GIC, other collaborators, and of course, funding agencies and yourselves for your attention. Thank you.
Many thanks, Paul. That's fascinating. Catherine, have we got any questions at this point?
Not at the moment, no.
Perhaps just one comment, Paul, so you don't get off scot-free. What about overexpression of glucosyl transferases? Is that something that's being explored at all?
Yes, one of the genes that we're currently looking at, which we think is the resistance factor, we believe can be modified by some of these responses are repressed naturally in the plant, and without modifying the plant through a GM approach, slightly altering the region upstream of the gene can alter sites where repressors sit. And so you can make that detoxification mechanism come on earlier or when there's less stress than the plant currently needs. So it gives it like a hair trigger. And so that's what we're looking at, is whether or not we can introduce some of these hair triggers to increase the sensitivity of the plant to potential attack before the fungus overwhelms it. And so it would then enhance its ability to detoxify this virulence factor.
Thank you very much. Obviously, it's a long road ahead to exploit all these approaches, but you know, may get there in the end.
One day, perhaps.
Yeah. Okay, so I think we better move on, as we're a little bit behind time. So now, my pleasure to welcome Aoife O'Driscoll from NIAB. Aoife is actually a molecular plant pathologist by training, but nowadays is more involved, if I've understood it correctly, in you know, potentially moving some of this fundamental knowledge into the agronomy and knowledge transfer in the arable and horticulture crop sectors. Today she's going to be talking pathology into practice. So welcome, Aoife. Can't hear her at the moment.
Thanks, John. I'm just going to figure out how to share my screen. And if you just let me know when you can see that.
We can see your screen, but it's not in presenter mode.
Perfect. So I'll just do that now. That okay?
Yeah.
Brilliant. Okay, great. So I was given a title for my talk of Pathology into Practice, and that's exactly what we're going to do for the next 15 to 20 minutes. So just look at what we've talked about over the last 2 hours from colleagues from UKCPVS and the previous talks, and then how do you actually apply everything we've heard when you're standing in a field in March and April and May and June, and you're an agronomist managing lots of different varieties, and you're trying to make a decision around what to do with your yellow rust. So any pathogen or any kind of host pathogen interaction or disease control strategy is based around 3 things. So the pathogen, as we've heard about earlier from Charlotte and Amelia and Nicola, looking at virulence and fungicide sensitivity. And also Paul talked about resistance and so deletes around resistance and fungicide requirements. But what we're really going to focus on in the next kind of 15 to 20 minutes is decisions around the environment and also the host or the variety. So these 3 things have to come together to form a successful disease interaction. So if we're thinking first of all about what are the factors that affect disease risk for yellow rust in particular. So you've got 2 sides to the coin here. You've got the risk of the crop being exposed to infection from early on in the season right the way through to harvest, and then also the ability of the crop to resist or tolerate infection when the rust actually starts to come in. And both of these will be affected by different factors. So obviously location, climate, previous crop, all your different cultural controls, and we'll go through these as we go through, and also variety choice. So how do you manage those risks as you're going through the season? And I suppose the first thing to think about is when you talk about yellow rust, we rely on a study that was done back in 2007 from Pete Cladders and colleagues over at ADAS, which looked at thousands of different fungicide-treated crops and looked at the epidemiology to find out what were the major factors influencing kind of yellow rust development. Everything from variety resistance to sowing date, number of air frosts, and the number of fungicide sprays. So from the context of managing yellow rust on the ground and in UK settings, the first thing you need to think about as you go into the season is the impact of sowing date. So we know very clearly that as you go into October, early November, into late November, that the challenge around managing yellow rust increases. So you're more likely to get infection, and your risk increases as you delay that sowing. So you take that and you think about, okay, well, I'm an agronomist and I'm managing lots of different varieties, and I've put some of these varieties here. So I've got some X-Days, some Graham, some Skyfall, some Crusoe, some Zayat, Champion and Dawson, and Skyscraper. And I've got lots of different sowing dates. So all the different farms that I'm looking after or managing, or maybe you're a farm manager with lots of different crops in the ground, all these different sowing dates. How the hell do I prioritize them for managing them for yellow rust? So that's a priority for you in the area that you're sitting in. So the first thing to do is think about, so first of all, sowing date. So think about when it was sown. So the further you go, late September, October, into November, going to late November, the further you push that, even with the resistant variety, that will increase your risk. So even for something like X days, which on the watch list is recorded as, say, highly resistant, and with the yellow rust rating of 8, if you sowed in, say, September, even late September, the risk will stay the same if you kind of sowed in an earlier or mid sowing slot. Pushing that risk into late November, even with a highly resistant variety, on the seeding tests and the adult plant resistance, will push that risk up a little bit. So you should think about prioritizing that for management. Where something like, say, Champion, where it's a good resistance, but it's late sown, again, you need to prioritize that. So how do you think about which ones to deprioritize? So again, looking at things like Crusoe and Dawson, which again are both highly resistant according to the watch list, the AHDB watch list, and have resistance ratings of 9. So you can probably deprioritize those. And this is a very realistic situation. If you're an agronomist, perhaps in say the east of England or in certain parts of the country, you're going to have a lot of different varieties, lots of different sowing dates. And how do you prioritize them? So first of all, you need to think about your variety. You need to look at the watch list scores to see how your variety will be affected by that those early yellow rust epidemics that might be arising after the winter. Then you also need to look at the resistance scores on the recommended list, and then also consider your sowing date, and try and consider all those factors when you're thinking about which varieties to prioritize in terms of management as you go through the season. The next factor to consider is what was the weather like over the winter? And again, going back to the peak ladders paper, so we know very clearly that the number of air frosts is a big driver when it comes to the its abilities to knock back the early yellow rust population. So basically, the further you go down in terms of the number of frosts, so anything from 5 days of air frosts where they're below -5 will knock back the yellow rust on the outer leaf surfaces, will also knock back the mycelium that is living in the leaf tissue itself. So a very basic rule of thumb is if you've had 5 days of air frost with -5 degrees, that will contribute to lowering the risk in terms of the amount of yellow rust present. It doesn't eliminate it completely, but it does reduce the risk. So again, another factor to consider on top of your variety, your drilling date, and your rating, is to look at the number of frosts that you've had over December, January, and possibly even to February in certain parts of the country. The next thing to consider then is the weather effects. So as you go through the season, so you go from February, March, and into April and May, what factors drive yellow rusts as you're going into the season? So we know, again, as I said previously, -5 degrees air frosts reduce survival. And with the other diseases, brown rusts, mildew, we have certain factors that we know drive disease epidemics if there's active rusts or mildew sitting there. So you need to consider those factors. And again, because of time, I'm not going to go through them in too much detail, but they're fairly well established what those weather effects are. And then I think this is an important one to spend some time on. So there's a lot of conversation at the moment around balanced nutrition and a healthy crop equals more disease-resistant crop. And it's a very complicated situation, and it can be very nuanced, and it's difficult to have a kind of a concrete debate around it. But there are a number of things we do know about different pathogens and different management strategies for each of those types of pathogens. So the first thing that we do know is that excess nitrogen, and specifically where you have fertile sites as well, these favour mildew and to a lesser extent rust. So this comes down to the amount of nutrition available in the plant, but also if you're on a fertile site and you have an early drilled variety, you can have a nice lush canopy, especially after coming into the season. And with some varieties that have a faster that go through their growth stages much faster. But for a lesser extent, that does affect rusts. And then the question around micronutrients, again, is one that is very nuanced, and doesn't have a very concrete answer to some of it. But we do know, for example, that boron, copper and manganese do play a role in improving disease resistance or susceptibility. So we know that, for example, a lot of crops kind of going into the springtime, they suffer manganese deficiency, and so that increases their mildew susceptibility. So most people out there will be applying manganese, and that will help improve the susceptibility to mildew. I think it's really important when you're having this conversation around balanced nutrition and crop health products, is to think about the trophic status of the pathogen. So we know, for example, that biotrophs, that includes the rusts and the mildew, they thrive on really healthy, living, well-fed plant tissues. So you can actually, you can overfeed your crops, and you can have what we call fat crops. And that's not good for things like rusts and mildew, where they really thrive on this really green, lush canopy. Whereas something like a nectarotroph, like a net blotch, for example, they thrive on stressed crops. So when you're having that debate around the healthy crops equals more disease-resistant crops, it's not that simple. Just because you have a well-fed plant with lots of micronutrients in there, it doesn't make it ideal for certain pathogens. And it's also very difficult to know what a balanced nutrition program is. Again, lots of information out there, but just maybe consider some of these factors when you're thinking about using some of these products for managing yellow rust. And it's also important to remember that if you're thinking about products that improve plant health, just remember that a good start is half the work. So you should have done half that work when you were drilling the crop itself. You sowed it into a good seed bed, you sowed it at the right time, into good conditions. You should be already halfway there in achieving a healthy crop. Some of these products might not be necessary if you already have a crop that's well established with a good stand and at the right growth stage for the time that it should be at. The next thing to think about is fungicide timing. So traditionally, fungicide timings are designed for septoria, and that's always the way it's been. And of course, the problem with that is the first thing is around latent periods. So we have very traditional T0s, T1s, T2s, and T3s. And that comes back to the latent period of septoria, which can be anywhere from 14 days, which they say it's probably up to 28 days. It's probably more than that, to be honest, probably 31 to 32 days in certain environments. But if you do have a very fast cycling septoria isolate, you can get up to 6 cycles in a season. And that's really where the traditional timings came from, along with the leaf layer emergence. Or for something like yellow rust or even brown rust, it's a much faster cycling pathogen. So if you have if you have the conditions that are ideal for a yellow rust infection, you can get up to 12 cycles in a very, very high pressure season. And for brown rust, it's even more than that. So you can get almost double the amount of cycles, again, if you have a high pressure season. So your crops are going to be at risk from yellow rust if you go by these traditional timings at T0, or you might even need a T1.5, depending on how bad the yellow rust epidemic is as you go into the season on your variety. If the yellow rust is cycling quite fast, you might need a T1.5. But then when we come on to later, we'll talk about product choices. You're really limited in terms of a number of different factors. So how many applications can you make of that product in the season? The total dose rate, these are both important for things like tebuconazole and STHIs. Growth stage limitations is another factor. How many days you might need to leave in between treatments, and of course, Nicola mentioned earlier around good frag compliance. So if you're applying a product for yellow rust, does it comply with the fungicide resistance management guidelines, especially for very susceptible for pathogens like septoria that do need really good compliance with the frag guidelines. Taking these factors into account, so bearing in mind that if it's a very, very high yellow rust pressure year, and you might end up having maybe 4 or 5 applications in a season. I know last year where there was some farmers got cut out with certain varieties where they ended up doing 7 applications in a season to keep on top of the yellow rust. What options do you have available to you in terms of fungicides? And again, this is just some guidance that we produce in NIAB, and this guidance is again based around septoria. So our fungicide timings are based around septoria management, and the guidance that this table that we've produced was built as well for around septoria management. So we're looking at the main products that we have, fungicide products that we have available to us. So Univoc, Revistar, Ascra, Latacera, older SDHIs, and then the older triazoles and Folpet as well. And this looks at the curative, protectant, and persistence in the field. But if you turn this around and look at it from a yellow rust point of view, this is probably where we are in terms of how the products fit for yellow rust management. So for rust, Latticera, which it remains the best product there for both protectant and persistent capabilities, and tebuconozoles remains the best eradicant. But we do have a number of other options there which provide good curative and protectant ability, as well as very good persistence, especially the newer products like Univoc and Revistar. But again, these are all going to be affected by the number of applications that you could do in a season, growth stage limitations, and also what else you're trying to manage in that season. So if you're in a very high septoria pressure area, or you're managing a quality wheat where you're trying to keep Fusariums and other diseases out of the ear, you have to take all those factors into consideration when you're planning your program. This is also some AHDB data which backs up what we've shown in the previous slide. So from the fungicide performance over the last couple of years, and it's pretty clear there that, as we mentioned in the previous slide, Latticera really is the best product there for septoria control, or not for rust control, sorry. Whereas other products like Pathogen, the Univoc, etc., they do give really good activity, but Latticera and Lattice Plus are definitely the best in terms of yellow rust management. But you can't deny, looking on the right hand side there, where you have straight Imtrex, straight Myrica, and the straight Pectiga as well, looking at their various responses there, that the Imtrex and the Myrica still giving good, if not moderate control. So that's why you have those mixtures with Univoc and Revistar. Is the extra chemistry in there, gives you that added boost in terms of yellow rust control. Okay, so what about further IPM approaches? So you've got the whole way through the season, you've planned your fungicide program, but we are in a situation, where we are thinking about other ways of managing diseases. So what else can you do? At least touched on variety mixtures earlier, and yes, they definitely can help. I'm going to show you some pictures later on from a trial that we did, a NIAB trial done in Hampshire this year. They can definitely help in reducing or delaying the development of yellow rust epidemics, especially in organic situations. You could also try intercropping. So we're looking at things like beans and wheat to help reduce the disease pressure by either dilution or acting as a physical barrier. And also grazing. So I call it a T-sheep instead of a T-knot. So grazing the crop down to a certain level to both get rid of the early inoculum and also to control your canopy. So if you've got a very fast developing variety that's gone through its growth stages and you need to knock back the canopy a little bit, grazing can be an additional useful tool. And yeah, where possible, they can be used as part of an integrated approach with all the other tools that we've talked about previously. This is just some pictures from a trial that we did down in Sutton, Scotland. So Lise mentioned about the variety mixtures work that they're looking at in Denmark, and how a lot of that work is based around feed wheat. And so we decided to look at it from a millimeat perspective. And so what we did was we had we had Zayat, four millimeat varieties, so Zayat, Crusoe, Skyfall, and Illustrious. And we looked at mixtures of all four together and then compared it to the individual varieties as a straight. We had them completely untreated, so no fungicide at all. A reduced rate program, so that was a third dose of Revistar and Univoc at T1 and T2, and then also what we call a full fungicide program, which is just basically a farm standard program. And I think this is really the message we're trying to get across with mixtures, is that it's not one or the other. It's about using all the tools available to you in an integrated program. So up the top left-hand corner here, we have Zayat. This is just straight Zayat, sold as a pure stand. It's fully untreated. You've got yellow rust heaving through the crop there. Now you can manage that Zayat fairly well with the standard fungicide program, just with tebuconazole at T0 and T3, and then a Revistar and a Univoc at T1 and T2. And that program managed the yellow rust very well. But you can also manage it by putting Zayat in a mix. And on the bottom two pictures here, you've got Zayat in a four-way mix with Zayat, Crusoe, Skyfall, and Illustrious. And you can see here it's either a reduced rate program or fully untreated. And the effect that putting that Zayat into the four-way mix had on the disease levels in that crop. So again, it's not about one or the other. It's not about you know, putting diet in a mix and not applying the fungicide. It's about integrating all the individual components together to help you reduce that inoculum and get the best use out of your fungicides in that crop. And then finally, just to sum up, it all comes back to assessing the risk. Everything you do, if you're on a farm and you're trying to manage multiple varieties, multiple sowing dates, and you've got a lot of things to do, because there's lots of other things to consider as well. It's not just yellow rust management. You just have to think about how are you going to manage the risk, and you have to go back to think about the risk of being exposed to infection. So thinking about your varieties, your drilling date, the weather conditions prior, and also the ability to resist that infection. So what's the resistance rating both at the seedling stage and the adult plant stage? That information is available to you, and what the ability to resist infection will look like, also to do with the weather patterns that we're going to see going into the year. So how do you integrate all those things? So you've got the monitoring and surveillance tools from the UKCPVS. You need to think about what your the things that you're doing on farm are going to impact on other agronomic aspects. So again, your drilling date, your variety choice, the number of varieties that you sow. So if you have a larger number of varieties, that causes its own issues, but also it's better in certain ways to have a larger number of varieties, because it helps you spread the risk and spread the load in terms of managing those. You have to match your situation to the risk. There is no blueprint, unfortunately, and there never will be. Every season is going to be different. Varieties behave differently according to all the different pressures that we put on them. And then, of course, fungicide programs or armory is decreasing. So you're going to have to be really, really careful and think about all the different aspects of disease control when you're thinking about how to design your programs. And the final thing was, I came across this quote on social media over the weekend where one of the farmers said he felt like he was an applicator and not a decision maker, because he wasn't thinking as much about how his on-farm decisions were impacting on his disease control strategies in the field. So take-home messages: try and be a decision maker. Put all the different things that we've talked about today around monitoring and surveillance, the agronomic impacts on farm, what Catherine's going to talk about in terms of resistance rating, put all those into play and be a decision maker, and don't just be an applicator. And that's it, and I hope I've managed to make some time back.
Well, thank you, but yes, we've gained a little bit of time. We have actually got a question here, which I think I'll just pick up immediately, because obviously the discussion on variety mixtures has triggered quite a lot of interest.
Good.
What were the yield differences in your mixed trial?
So I will say that we had other mixtures in there as well, and the mixture didn't necessarily out yield the individual components in all the different fungicide treatments, but that's kind of the point. The whole point of mixtures is not that it's going to ever give you more yield than the individual components, but it's about helping you manage your risk that way. So being average, it was pretty similar. The mixture was pretty similar to the individual components, but you're managing your risk in other ways as well. So things like weed control, disease control, reducing your fungicide inputs. So in terms of margin, there was probably more an effect in terms of margin because you're reducing your fungicide spend. But yeah, we didn't see a big yield improvement in the mixtures versus the straights. I'm happy to share that data separately if anyone wants to contact me about it.
Okay, thank you very much, Aoife. That's very helpful. Okay, I think in view of time, we ought to move straight on to our next presentation, and in fact, the final presentation. And this is Catherine Harris from AHDB. She's Senior Crop Protection Scientist for cereal and oilseeds at AHDB, and she's responsible for the recommended list ratings and fungicide performance curves. So obviously in an ideal position to tell us a bit more about the recommended lists. Over to Catherine.
Thank you. Sorry, I'm just trying to organise all of the things on my screen. Can you see my screen with the PowerPoint in presenter mode? Okay.
Yeah, we can see it.
Okay, thank you. Great. So yeah, nice to be here today. So as John already said, my name is Catherine Harries and I work in the IPM team at AHDB. I'm responsible for the recommended list disease ratings, and I manage the UKCPVS project. So Charlotte's already mentioned how the UKCPVS provides isolates for RL inoculated trials, which feed into the disease ratings. But the UKCPVS can also help us understand anything unusual. So, for example, if a previously resistant varieties are getting high levels of disease, we use that information to validate and inform the changes. So, for example, a few years ago, we decided to calculate the ratings just using 1 year of data, as it looked like there'd been a shift in the yellow rust population. But today I just wanted to talk about two things which are relatively new additions to the RL, which is the inclusion of the young plant resistance and susceptibility information in the RL booklet, and also the watch lists, which is a way to make use of the RL data to help with varietal monitoring and management. I then wanted to go on to talk about an evaluation of the project that we've just started, and hoping that some of the people here today will volunteer to be part of it. So previous speakers have already mentioned this, but resistance to yellow rust in wheat operates differently at different growth stages. So there's the young plant or seedling resistance and the adult resistance, which kicks in by growth stage 55. And sometimes a variety is susceptible at the young plant stage, but resistant at the adult plant stage. So at the moment, the RL ratings are based only on adult plant data for yellow rust. So that's all the data that's collected after growth stage 55. But farmers and agronomists are having to make spray decisions throughout the young plant stage up until T2. So resistance at this stage is just as important for them. So that's why, for the first time this year, we've included an extra line in the RL disease ratings. Here you can see the wheat disease ratings, and it shows which wheat varieties are resistant or susceptible to yellow rust at the young plant stage, with an S for susceptible and an R for resistant. And this categorisation into R and S is based on a combination of UKCPVS and RL data. So this table shows the UKCPVS data with the variety, RL, adult plant resistance, so the main RL rating in the previous year and the current year, and then also the seedling stage resistance for both years. And as mentioned earlier, this is categorised using 5 isolates, and a variety is classed as susceptible if it's susceptible to any one of the isolates at the young plant stage. But because isolates used for testing are a subset of the population, actual field performance might vary if there's other isolates out in the field not captured by the UKCPVS. So that's why we validate the UKCPVS data with RL field data from before growth stage 55. And you can see here, I've removed the variety names in the data, but this shows the 4 trials that we got data from in the RL in 2022 from before growth stage 55. Oh, have you lost my screen? No. So yeah, on the right here, we can see the resistance or susceptible categorisation from the UKCPVS. And you can see that all the varieties that are classed as resistant consistently had disease levels of below 5%, except for this one variety, variety 7 here. And because this variety had more than 5% disease at one site, we reclassified its seedling status as susceptible. And this was just to make sure we're capturing everything that's happening in the field that might reflect infection from isolates not yet included in the UKCPVS testing. So that's how we put that table together, and we'll plan to include that next year as well. So moving on to the watch lists. The yellow rust watch list was first introduced in 2021 using the data that we collected in harvest 2020. And this year, for the first time, we've introduced a brown rust watch list. And what the watch lists aim to do is to reveal varieties that have performed out of line with their recommended list ratings at some sites in that year. So that means it can highlight varieties that might be vulnerable to falls and resistance, and the aim is to focus crop walking and management. So it uses the RL data, all of the data that goes into the main RL ratings, but then once we've collected all of that, we use the data from the UKCPVS adult plant trials to validate what we see. And this is how it's displayed in this table on the right. It shows the varieties ordered by their performance at the 3 sites where each individual variety performed the worst compared to how it normally performs. And then alongside this is the main RL rating. So a variety that is low down in this section of the table but has a relatively high RL rating shows that it's performed worse than normal at between 1 and 3 sites. So the easiest way to visualize this is in a graph like this at the bottom. And this shows the main RL rating along the bottom and the position on the watch list along the side. And varieties towards the bottom right of this chart are increasingly likely to perform worse than expected in some locations in the following year if the race in question increases in the following year. So how well has the yellow rust watch list worked in the last few years to protect varieties whose resistance might be being eroded? Well, we can look at this by comparing the difference in a variety's average performance against yellow rust to its performance at its 3 worst sites. So that is the information that's provided in the watch list. And this is shown here for 2020 on the y-axis, and with how much, so we compare that with how much its RL rating fell in the following season, which is shown on the x-axis. So a smaller difference on the y-axis shows the varieties performing consistently and as expected across all sites, whereas a larger difference shows that the varieties performing worse than average at between 1 to 3 sites. So a larger number on the y-axis shows a potential breakdown. So what does this show for the from the Harvest 2020 watch list? Well, you can see there seems to be a correlation between the 2 factors. So overall, where the difference on the y-axis is bigger, the fall in rating from 2020 to 2021 was bigger. And this suggests that the watch list can give an indication of changes in rating in the following season, and shows it can work well. But there are lots of factors to consider when making this assumption. So for example, we can see there are some varieties where there's a large difference on the y-axis, but no change in rating, these ones here. And also some races might be a problem one season and then fizzle out the next. So this variety here, for example, is an oddity. And what happened here is that in 2020, this variety overall performed fine. It had an overall RL rating of 6, but at 3 sites it performed worse, giving it a rating of 4.6 just based on those 3 sites. So normally this might suggest that a new strain of yellow rust virulent on this variety was emerging, so we might expect further drops in subsequent years. But in this situation, the following year, the variety actually performed better, giving it a rating 0.4 higher than that in 2020. So it had a rating of 6.5. And what about the prediction success for 2022 from the 2021 watch list? Well, there wasn't so much to see in 2021, and there were fewer mismatches in performance, with only a handful of varieties dropping in rating. So any possible new races picked up in 2021 for whatever reason didn't seem to cause a problem in the following season. And this just emphasises that what actually happens the following year can't be predicted, but the tool can still be used to help focus monitoring and management. So what about the watch list produced from the Harvest 2022 data that might predict performance this coming season? Well, you can see here for yellow rust, the relationship between performance at all sites on the x-axis and performance at the worst 3 sites for each variety on the y-axis. And if you remember, varieties towards the bottom right of the chart are the ones to look out for. And you can see that overall, all varieties are performing as expected. If you wanted to pick any out, you might pick out these two, which have RR ratings of 9 and 7, but seem to have performed very, very slightly worse than average at some sites. And this was from slightly higher disease levels in some Norfolk trials, but it only related to a 2 to 3% increase in diseases in yellow rust compared to the other sites, so it's not really anything to be worried about at all. So the good news for 2023 for yellow rust is that these 2022 data aren't predicting any potential problems, but obviously to emphasise, anything could happen, so please do continue to monitor all varieties. For the first time this year, we have released a brown rust watch list, and here the situation is slightly different. So as you can see, most varieties have performed as expected. This is the same chart as I've just shown for yellow rust, and you can see for brown rust there's a good correlation between the main RR rating and its position on the watch list. But this variety, which is Theodore, has a main RR rating of 8, but as Amelia spoke about earlier, it performed, so the Devon site that Amelia was talking about was actually our RL trial site. So we have, although we don't have the isolate for the UKCPVS, we do have the data. So it performed out of line at this Devon site, where it had relatively high levels of brown rust at 19%, which was the second highest level only after Crusoe in that trial. And this result suggests there could be some change in the pathogen population, whether that's a new race or re-emergence of an older race, which we should look out for in 2023. But the significance of this isn't known. So again, continue to monitor all varieties and send in any samples to the UKCPVS if you see anything odd going on. So that's everything I had to present on the data, but finally I just wanted to bring your attention to the fact that we're going to be running an evaluation of this project. So we're always looking at what practical difference the projects that AHDB fund make to our levy payers and the value the levy payers get from them. So with the current term of the UKCPVS project coming to an end, it's a really good time to review this project and make sure the levy funds are being spent appropriately. So we're running this evaluation, and we have an independent consultant called Paul Teven, who's going to be speaking to breeders, agronomy companies, industry experts, etc. And we really need to have some volunteers to speak to him. It will just be a 20-minute telephone call, shouldn't be too onerous. So when this webinar ends, you'll be presented with a feedback form. And if you are happy to be contacted by him, please do say so, and then we'll be able to get in contact, and that'll be really helpful for the project. So thank you in advance for that. And just as this is some of the, so Paul Teven, the independent evaluator, will be looking at different options of what the project might look like in the future, and we'll evaluate them against some different criteria to get the results. And some questions we'll be asking are things like, do we understand it? Do we use it? Does it make changes about decision making? The cost benefit analysis of the program, and things like that. So yeah, please do say if you're happy to be contacted by Paul. That would be really helpful. And thank you. I'm happy to take any questions, but I'll just need to go and find them. Maya, can you stop me sharing my screen and then I'll be able to look at the question? There are no questions, so I'll hand back to John.
Okay, well, thanks very much, Catherine. We recovered a little bit of time there, so we're not too late. Thanks very much for your coverage of the recommended list, which I found extremely interesting. I suppose at this point, I'm expected to make a few closing remarks. First, I think actually I ought to have some thanks here. I'd thank all of the speakers for doing a good job, I thought. I always think doing stuff online is more complicated than when you're sitting in a room and you can see what's happening all around you. So they've done very well to keep largely to time, and also a very nice range of different topics. So thank you to them. One thing that struck me is the complexity of agricultural decision making. And at this point, I'm quite pleased that the only decision I have to make now is what I'm going to have for lunch. Also, I should thank Mia Koteka, who helped us with all this, the technical side of this presentations, and also all the attendees, who unfortunately I can't see, but I hope you found it useful and interesting. And please do remember to fill in your feedback forms, please, because the information in those is always useful, and you know, hopefully will make for better meetings in the future. And we do have our final slide there, I think, which is really from AHDB, I think. And thank you to everyone. All the best.